Friends on microbial pathogenesis and recent advances in diagnosis of Omicron. I feel a privilege to welcome today's speaker. I would like to invite Professor Sai Gopal sir, Vice Chancellor of Cluster University Kurnool and Professor of Virology Department, SV University, Tirupati. A warm welcome to you, sir.
Thank you.
Over to Mercy.
Thank you, Dr. Shanti Priya, ma'am. Good morning, everyone.
Yeah. Good morning.
Good morning, sir. We welcome you to the webinar. To introduce the speaker, I would like to invite Dr. N. Kishore, sir, HOD, Department of Microbiology, Palamura University. Over to you, sir.
Mercy, unmute yourself, Amma.
Can you hear me, ma'am?
You can continue. Volume mentioned.
Yeah.
It is my immense pleasure to introduce our today's speaker, Professor DVR Sai Gopal sir. Professor DVR Sai Gopal sir is presently working as Vice Chancellor of Cluster University at Kurnool. Earlier, he worked as Professor and Head Department of Virology at Sri Venkateshwara University, Tirupati. He has 32 years of teaching experience at PG level and awarded 22 PhDs. His area of research is molecular characterization of viruses and development of diagnostics. He worked for development of Center for Viral Diagnostics, antiviral studies of active biomolecules by in vitro methods with a goal for development and commercialization of viral diagnostic kits with industry linkage. Dr. Sai Gopal acted as coordinator DST Purse Center at SVU, regional coordinator for AP Academy of Science, Andhra Pradesh. He was also nominated as Chairman Board of Studies for SVU Pharmacy College, Board of Studies member in Microbiology and Biotechnology of other universities of AP, Karnataka, and Tamil Nadu. He was appointed as Indian Coordinator for Erasmus Mundus BRAVE program of European Commission. Dr. Sai Gopal received Best Teacher Award of AP State in 2017 by Government of Andhra Pradesh, AP Academy of Science Fellow Award in 2015, Excellence Award in 2015 by KL University, Guntur. He also received AP Best Teacher Award in 2014, Lifetime Achievement Award in Applied Microbiology, Fellow of Association of Biotechnology and Pharmacy. He received grants for many organizations, many research funding agencies such as UGC, DBT, DST, Defense Research and Development Organization, DRDO, Erasmus Mundus. He visited abroad by invitation to NCRM Nichi, Tokyo in 2018. He visited Athens, Greece under Erasmus Mundus BRAVE program in 2017, Tuskegee University, Alabama, USA as guest faculty, and also as an international trainee at IRRI, Philippines. Dr. Sai Gopal had an international collaboration with NCRM Tokyo, Japan, Athens Agriculture University, Athens, IRRI, Philippines, Delhi University South Campus, NCRM Chennai, IRI New Delhi, NIRI Nagpur, SVIMS Tirupati, SV Veterinary University Tirupati, and Frontier Technology Laboratory Tirupati. He published more than 155 research publications, out of which 112 are international and 43 are national. Papers were published in Scopus indexed journals with a total impact of 58.36. He published 10 textbooks and 4 laboratory manuals as co-author. 9 Indian patents are registered in his name, out of which 2 are under commercialization process. He organized 6 international conferences, 5 national conferences, and 15 national and regional workshops. Dr. Sai Gopal is actually engaged as editorial member to Journal of Clinical and Scientific Research. He is also a reviewer to International Journal of Virology, Journal of General Virology, Indian Journal of Virology, etc. He is acting as an institutional committee for stem cell research and therapy in ISER Tirupati. He is a member in SVU Bioethical Committee. He is a member in Institutional STEM Research Committee of SWIMS Tirupati. He is a chairman of Institutional Animal Ethics Committee of Sri Vidya Niketan College of Pharmacy and Seven Hills Pharmacy College. With this short introduction, it is my privilege to welcome Dr. Sai Gopal sir to deliver his lecture. Thank you very much. Over to sir.
Thank you, Doctor. Are you able to hear my voice?
Yes, sir.
Yeah. Thank you for inviting me for this two-day international e-conference on microbial pathogenesis and recent advances in diagnosis of Omicron. So I'm happy that yesterday I heard some of the few talks by Shermistra and other people, and also thank you for inviting me, Dr. Shantipriya. And also I highly appreciate Dr. Kishore has narrated my brief biography. With this one, I'm happy to associate in this international two-day conference, which is up to today on related to the Omicron, which is playing a very big role in the global scenario, and also one of the variants in that. So with this one, I'm highly thankful for the Dr. Shantipriya, and I'm happy to see yesterday Honorable Vice Chancellor and Registrar and all this, they have given a lot of scope for the virology importance in the today's scenario and how they can change the worldwide, because there is a proverb is there. So the bullets and then the nuclear bomb cannot change. The virus can change total global scenario, where it is which is invisible, which can make the things visible in society and industry and the human welfare, animal and plant welfare, all directions, multi-directional. The effect is there by using the COVID-19. So with this one, I can highly thankful for the organizers giving the opportunity to share whatever little knowledge I had in this COVID-19, because we did some work on this, and especially diagnostic part and all. I will share with this. Thank you for one and all. Can I share my slides? Permit me?
Yes, sir.
Are you able to see the slides, Dr. Mercy?
Yes, sir.
Yeah, it's okay. Is it clear? Full screen has come?
Yes, sir.
Okay, so this is the topic what I selected is the recent trends in diagnosis of the Omicron, because everybody knows. So this is where the COVID, which is belongs to coronavirus family. So especially there are sister families are there. So like which is influenza also comes under, and that already we face a lot of problems with the swine flu, and also with this avian influenza and human influenza, because the viruses which are playing very important role, especially the RNA viruses, especially the positive sense single stranded RNA, linear, and there are all of this is the viruses as a model for genetic diversity. You don't see the genetic material any other microorganisms, so including fungal, bacterial, and parasites. The RNA is genome, and also in the DNA also diversity is there, double stranded DNA viruses, single strand DNA viruses, and then single strand RNA, double strand RNA, positive sense, negative sense, like that. There is a lot of genome diversity is there in the viruses. So due to that genome diversity, the viruses which is going to wide host range infecting several organisms, especially the zoonotic virus which is coming from animal to the human. And recently in the last international conference where the virology, they had a discussion, are the plant viruses are going to infect the human. Already the evidence was there, the animal viruses are infected to the humans. Already cycle has started. But in the future, the plant viruses are there. Example, Rio viruses. So the Rio viruses, which are exactly, they can multiply in the plant, same way multiply in the humans also. That's why the bridging, the phyto-Rio viruses, which is bridging between the plant and the human viruses, probably in the future, they may come into the human cycle also through the plant material and some different way. Okay, that is a different story of that. But here, the corona, which is, I think yesterday has a lot of discussion was there on this, because the spike, because this enveloped virus, you can see the spike proteins, which are glycoproteins, which are playing very, very important role, which can be bind to the human cells, and the human cells will go and then tag or entrap this by using receptor mediated mechanism. So these spikes are, which can be, lot of variable is there. So you know that the viruses which has the mechanism calling as mimicry. So when the virus enter into the system, immediately the antibodies, primary immune mechanism started, later the overrided, then coming to secondary immune mechanism where antigen specific antibodies will be raised in the body. So when the time the spikes will go on change what you can call as the structure. So due to the structure change, the antibody is unable to bind to the epitopes of the surface antigens of the virus. So what happens again, the antibodies change, again the spikes will change. That is going on changing, we can call as mimicry, that is calling in the immunology, people says antigenic shift and drift mechanism. Like that, the coronavirus has the very high frequency of the changing in their character, especially the spike one. That's why I put this, the spike is playing very, very important role, because these spikes which are changing depending on this encoding of the genome only. It's because all of you have to remember that the genome which can change a single nucleotide, which change the single amino acid or the multiple amino acids in the expression. So here you can see that, so a lot of mutations are going on because the spikes are playing important, which can play to bind to the human chain. All of you, everybody knows now, I think the virology became very popular, that the layman can speak about the what you can call as internal detail of the virus. Earlier nobody knows, simply says virus, HIV, AIDS only they know, and influenza. But now the layman can speak what are the receptors, what are the spikes, how they can there, what is the AC2, that layman people now understand the viral changes. And also the corona has totally given a lesson to the common people about the virology. That is, I think we can hear, the layman can get it, all the information. So here you can see timelines of the virus variants. I hope the next speaker, I think Rukhsana, I think she can speak about variants and so vaccines. I think I briefly touch here, I will go to the diagnostic part of this. So the timelines, you can see the variants concerned, the viruses which are going on, especially people scared about the different variants like beta, gamma, delta, and now Omicron. So these variants concerned, because these variants which are changing in their what you can call a structural configuration, I will go to that. So you have, all of you heard about different variants, beta B1351, beta 1617, like that. These are the what you can call as like the structural variations. So here the WHO is identified depending on their structural configuration of the molecule, especially molecular physics people, they know that crystallography analysis, how they can change. So they put the name, different names like alpha, beta, and then the delta, gamma, and epsilon, like that. These are going on about the changes of that. So the Omicron, you can see that the now is going on, which can be, so the spike proteins, you can see that, so these mutations is very high. And then the prevalence also, you can see the sequence is now studying, already it came out. So because this structure, which have a lot of mutations, and then they got the different variants in that. So the side which I can show that high number of mutations can formed because to the highly divergent in their what you can call a genome regions, around 26 to 37. I think there are now the 32 mutations are spike protein identified in the Omicron, especially recently. So here you can see that put the phylogenetic relation in this COVID. So this is entangled between the different, I think, pang and then bat, pang and back, where you can see that between that. So that's why this is a sequence homology. You can study about the phylogenetic analysis of this RNA. So what happens? There is a middle position occupied. So that's why they can easily mutate either that side, at bad side or other side. They are going on changing in their mutants and make different variants, especially in the beta domain of the phylogenetic domain of this. Now, I think this briefly I put it to understand the students, young researchers who are involved in the research of virology. So this is, if you want to study any change in their, especially in the surface, crystallography, the molecular biophysics people, they know the changes in the crystallography analysis, X-ray, diffraction studies. You can see a single beta pleated sheets, alpha helix, changes in their configuration. Then overall, it can change into their what you can call as the structure of the virus. Because this is a trimer, you can see the trimer which can make, where you can call triplex, the virus structure will change automatically. What happens, the virus which can characters also, and also clinical symptoms, subclinical symptoms, binding, and their what you can call as efficiency will change here. So here you can see carefully the histine, cysteine, cysteine, and these amino acids which can, so a single position of the what can change amino acid change in their what you can structure, especially in the beta pleats or in the alpha helix, where you can see that a rotational symmetry will occur, the virus structure can be changed. So this X-ray crystallography analysis, which can name the what type of virus, what is the what you can domain, the alpha domain is the beta domain. The crystallography, they says the structural variation in the virus structure. So that's why recently a lot of variants has come in. I think all of you, I think the students might have know that. So viruses, they have a lot of variants, mutants, and then you can call as the pathogens, and then you can call as different type of serotypes and the isolates, and there are different names are there. Sometimes it may be you have to understand those things. The pathotypes, serotypes, and then what variants, and then mutants and isolates, strains. These are the different words generally frequently used for any microbial organism. So you should be very clear that what is the strain, what is the mutant, what is the isolate, what is the serotype. But here, most of this COVID, you can see the variants are there. Within the variants also, the WHO has given different naming. That is what you can call as variant being monitored, VBM, VOI, variants of interest, and variants of concern, and the variants of high consequence, VOHS. So these which can designated, so on the viruses, depending on their what you can call as the character, how they can designate it. VOI, generally you might heard VOI and VOC, VOHS, VBM. So the COVID-19, which started as a VBM, now it has gone to VOI, VOC. So I like that variants as designated, and then these variants can be monitored by under the what you can study of the molecular analysis. So this is a picture where you can see that the lot of variants are identified, first record and place, and then the country and all. I think all of you know there is a symptomology, also variants, what you can call as clinical symptoms, subclinical symptoms are variant depending on their type, and that's why the vaccine efficacy and severity of illness, and then what is the transmissibility also variable in their see that you might have seen last 2 years the symptom variability is highly variable, starting from first one to now Omicron. You can see a lot of variation there, their symptoms, that is depending on the virus which can mutate it. So recently you can see that the WHO has designated. So this is a variant concern, the Omicron. So variant concern, which is B11529, because this Omicron, which is a lineage with the other types of the variants, B11, especially BA1, BA2, and BA3. And here you can see that 26 to 32 mutants are they identified in this Omicron. And then also when the mutants are changed, the diagnostics also will be changed. And once the diagnostics changed, because depending on the identification of the antigen in the body. So that's why the tools also should be changed to diagnose the viruses. So in this concern, the WHO updated every time. So this is at 21st January 22, where so they have given the recent how the variant can have their sequence and how they can summarize what are the risks and how they can identify what are the changes in their diagnostics, how they can useful for vaccine preparation and all. So this is what you can see. The WHO has given very clear pictures, the symptomatic suspected cases and suspected outbreaks and asymptomatic, how to diagnose the Omicron. So there are different versions are there in this, especially the antigen detection will be there, and rapid detection where with negative antigen negative and antigen positive and high clinical suspicion where you consider to with the amplification test will be there. So like that, there is a known case which is S or positive or negative like that. So here you can see these suspected symptoms, symptomatic one and a symptomatic one. So the lot of confusion is there in the especially diagnostic part of the viruses. So when we are going to clear, so the virus diagnostics, so there is a sometimes you can get the false negatives, sometimes you can get the false positives. So those you have to what you can call identify, and without the ambiguity you have to diagnosis. So that's why it is very crucial role that it plays about the diagnosis of the viruses. So generally the WHO has given so 3 models of the diagnostic tools. One is nucleic acid amplification test. Another one is the antigen detection by using a rapid diagnostic test. Another one is antibody testing against the virus. So these, which can be 3 are the what you can call as gold standard methods, which can be proposed by WHO. But within this, each one, there are different methods as standardized by different companies and different countries. They are using different platforms for the diagnosis of the viruses, starting from the nucleic acid amplification to antibody testing. But here, the antigen testing and antibody testing are crucial because you have to know the window period where you can see the virus. Sometimes the virus which moving from 14 to 23 days window period, sometimes they can act as a people symptomless carriers, some people which can be which acting as carriers. So like that, so when we are going to diagnostics, so these are you have to keep in mind, and then you have to do a lot of what you can call systematic diagnosis of the viruses. So if you can see the whole timeline of the coronavirus, where you can see that the detection of the RNA, I think all of you might have known lag and logarithmic, and so the what you can call as slope. Here you can see threshold of the infectiousness antigen. So in general, it may be antigen 7 days, but actually it starts after 7 days. The immunoglobulin, primary immunoglobulin, IgM, IgG, they will antibody response is there. But it sometimes always this may not be true, because what happens sometimes the window threshold of infectiousness will be prolonged, little bit longer period, and there is no strict like 7 days, but in the average it will be there, and you can see the viral load will be there in that period, threshold period, especially period of infectiousness, and antibody response you can see. So here I can understand this in the transition zone, transition period is very, very crucial. The patient generally will come. So then you can say this is a shifting is there, antigens which can be declined and antibodies are increasing. Then here the diagnostic playing very, very, very important role, because this transition where shifting of the antigen detection to the antibody detection. Like that, there are limitations are there in diagnostics. One is you have to detect the viral load, number of copies, depending on the sensitivity of the test, and the number of the immunoglobulin titer is also very important when you are going to diagnostics the antibodies and antigen in the tools. So here you can see there are the algorithms of different testings as given by WHO, asymptomatic people, how you can see that this is going on. So the patients, different flow chart has given, which can be with negative, positive, and negative. If the positive is there, what you have to confirm. If negative is there, what you have to do it. So like antibody test, if clinical suspicion. So like that, there are COVID-like symptoms are there, COVID symptoms are there, and then asymptomatic individuals are there. So like that, they have given different the flow charts to understand. So they rule out their positive have come as negative has come. Then it can go for the what you can call as a systematic screening of the sample by using the different tests are there. So, but here, for the understanding of the students and the researchers and researchers, I would like to know what are the methods which can be, these are the methods which can use direct detection by electron microscope, and then direct detection of the immuno capture, what you can call microscopy, where you can see the decoration of the virus particles and the, especially the antigen, and then you can study understanding of the virus structure. And these are the old methods will be there. Immunodiffusion test, which are still holds good in the laboratory. And these are the other methods which can be using for that. So these are all old methods. These are, but here recently, the old basic methods also coming into picture, because people say these are the old traditional methods, but still these methods are useful. Example, I will tell immunofluorescent antibody. So this is very, very useful nowadays. People, we can take the sputum samples, or you can take the what you can call as in the lung samples. So then you can take a smear and put it the labeled antibodies for that. And then you can see that the beautiful, see that the samples which can be fluorescent, and you can use by using the immunofluorescent detection method, both direct and indirect methods. And the radioimmunoassay also same. So with the low level antigens you want to detect, you can, sensitivity will be high. So people, but due to the some of the radioimmunoassay methods, due to the what you can call as radiation and all, people are avoiding that, but still it is very more sensitive method is there. But this is indirect methods are there, where the ELISA, everybody is now talking about the ELISA, dot ELISA, and then so the what you can call as the liquid and the plate ELISA like that. But these ELISA are still old, but people are using enormously for the enzyme linked immunosorbent test by the different indirect ELISA to detect the antigen, to detect the antibody, sandwich the antigen, and then you can go from competitive ELISA like the different methods are using, but still these methods can be used for the plate. Now it moved to the membrane. So now I think the membrane ELISA has come picture of the dot blot. You can number of samples you can collect, and then you block it, and using these chromogenic antibodies, the chromogenic substrates, by using antibodies you can do it. And then this is the luminescent. Now the people are using a luminescent assay method, where you can see the by using the luminol within milliseconds, the flash will come, you detect and capture that in the luminometer. So this is the SARS has recently has worked out. It was well success. The very low log values are there, and also you can detect by the difference between the healthy person, non-SARS person, and then SARS persons, where you can see COVID persons, where you can difference will be there. In this way, you can differentiate. So the variation in the viral diagnostic with high sensitivity will be there. So this is the what you can call as a immunoblotting. People can run, and then you can use the membrane transfer and all. I think the students know that the dot blot assay and all. So this is the method what nowadays people are, all the rapid diagnostics they are going on, especially lateral flow diagnostics. Now the kits are coming, what you can cassettes. So you can put the cassettes, which is a negative control, positive control, and control like that. You can put the sample of the nasal swabs by suspending into the transport medium and put it, and then people are identify and easily by lateral flow. This is nowadays is very popular method, what you can call a tri-dot method, dot method, and lateral flow. See here the lateral capillary, you can see that either you can detect the rapid diagnostic. RDT is very cheap nowadays. In the Omicron and the COVID, people are using the rapid diagnostics by detecting these what you can call as the antigen antibody detection by using the So the antibodies, and then you can see the flow, human anti-SARS-CoV antibody, SARS-CoV antigen, and you can tagging, and then the flow will be there. So control will also be there. So like that, the rapid diagnostic test, which can be used RDT for detection of the antibodies and also the detection of the antigens also. The simple method where if there are mixed samples also there, if there are 2-3 are what you can call as variants are there, like influenza, you can see how they can monoclonal antibodies, they can use it. And then you get a single shot, you can see which type of what you can call A or what you can call as which type of thing you can strain also, you can identify it, because, but you only thing is limitations here are the monoclonal antibody should be specific for that, what you can call as the strain or the mutant or the variant will be there. So like that, this is a lateral flow, which is very popular. Now people are using enormously for these rapid tests, but there are some of the usefulness. These are the rapid test of the membrane based or the fluorescent based, which is what you can call serological antibody detector based one, which can be cheaper but low sensitivity. And then sometimes, so it is difficult to, if the virus load is very low, difficult to detect the antigen, or the antibody titer is very low, it is difficult to detect the antibodies. That's why sometimes these serological techniques, which will be failed, sometimes it may give confusion, and sometimes it can give the negative impact on the diagnostics. That's why the serological now people are slowly avoiding, but only the suppose example I will tell you, if there is a transition period, the antigen shift into the antibody in the body system. So the techniques are very, very crucial, because the diagnostics playing important. That's why people are identified advanced diagnostic technology, which can be used by using different type of molecular based system. Especially these are the molecular based approaches are coming for viral diagnostics. Already they are using now the COVID, which is now become very popular, these techniques after the coming over this epidemic pandemics. So the polymerase chain reaction, everybody knows that is a very, very basic one. And nucleic acid specific hybridization test, loop mediated isothermal amplification, real-time PCR, and molecular beacons and array technology, RFLPs. These are the basic molecular tools which are using people for diagnostics of the viruses. But recently, after coming the Omicron, there are a lot of variants has come in the PCR-based diagnostics. So this PCR-based diagnostics, so what happens, the earlier olden days, generally they use for DNA viruses. Now, these techniques are now diversified into different type of tools. So now it's coming into RNA also. Within this, there are a lot of what you can call variants are there in the PCR. Now the Omicron, recently the European Commission, so they have designed specific type of what you can call as primers, in silico designing of the primers, and then the specific target of that, what you can call as amplicon, can be identified. So the efficiency will be more, and also the rapidity is there, and also the strainal, or you can call as the variant, highly specific variant can be diagnosed. That much the sensitivity and also the accuracy is there, and also repeatability is there in that. So that's why now the European Commission has come into the picture that there are a lot of the efficient methods which has designed by using different type of the what you can call in silico designing of specific primer, and also you can design the mixed primers, and you can detect the what you can multiplex also there in this. This is the now recently has given on that. So in the variants which can be there, I am not talking about the DNA amplification variants. This is because the virus is RNA. I put this RT-PCR and then IC-PCR and nested PCR and multiplex PCR, which are the models which can use for diagnosis of the different variants are there. So RT-PCR, everybody knows that reverse transcription PCR, where the RNA will be there. RNA can make into the cDNA template, and then it will go to oligo DTs, and then random primers will be there. Or you can see the oligo primers can be designed for these viruses. And then, so here, especially the crucial in the primers design is very important. And then the steps what are involved is very important, because you have to standardize the timings, because denaturation time, because each and every virus is not the same. They are going to change in this one. So the Immuno Capture Pcr also very important here. So the PCR tube will be coated with the specific monoclonal antibodies, and then the antibodies which can be bind the, you can collect the sample from the sputum or the nasal, put in the tube, it will go to bind to that antibodies, and antibodies will be trap the antigens with their very, very low. Then the Immuno Capture PCR, where the after the you have the disruption of the virus, then the RNA can be come out, and then you can use the DNTPs, and you can use the different primers, and then you can go for the amplification of this. So this is the what you can see there. Nested PCR is another one, where if you can want to test detection of virus with very low titer with infected one. So the nested PCR, you can see first primer, second set of primers, both will be used, and these target amplicants can be studied, and these which can be used very, very successfully. Now, the lot of variants are coming in the COVID. So the gamma, delta, alpha, and then the omega and all. So here, so if you want to detect these variants by using the multiplex PCR, you can design the primer specifically. Then you can use those primers into that. So multiple primers, and these primers can be targeted into different, so what you can call as the variants, and these you can use at a time. So in the single shot, you can detect either it may be the single strain, single variant, or multiple variants are there in the system. In the olden days, what they did was they used different viruses by using multiplex PCR. Suppose example, I will tell you. So we use a lot of medical legal cases in virology. What we did, suppose the person who has affected with the dengue, Japanese encephalitis, so like the chikungunya, 3 viruses, because the mode of transmission will be there, clinical symptoms almost will be there. So when you want to make the differentiation, so then you can collect the sample and do the multiplex. You can detect either it is a person is suffering with chikungunya, dengue, JE. In the same way, we made a designing of different PCR methods for the cervical cancer also. We detect now these Omicron, especially the COVID also, we can use different variants by using the multiplex. But here, the crucial step is to design the primers depending on the type of the what we are going to detect into the multiplex. That is very, very crucial, so that it can be detected here. I think yesterday, I think I have seen your registrar has narrated. So these different levels of the COVID level detection, especially CART values, how they can low titer will be detected, like that quantitative analysis of viral load, the limitations of there in the viral load. So these limitations also will have to overcome. And this is now earlier olden days, there is simple simulations are there. Now there are a lot of software has come. Automatically it can identify, and then it can be detected the load of the virus with very, very high level. So with accuracy will be high level. That much software has come into that. So now the restriction RFLP is also coming into picture, how this genome can be restricted and fragmentation. Then you can detect the single mutant. Now I think you can see the viruses COVID. So the single nucleotide change, which can change different type of what you can call as epitopes, especially so in the spike proteins. So like that, the people are identifying those changes easily by the restriction mechanism. And the another mechanism is the nucleic acid specific hybridization. This is also, you want to study the nucleic acid hybridized based principal binding studies specific to the specific nucleic acid you can targeting at, and you can do the radio labeled probes and non-radio labeled also there. NASH is one of the I think almost WHO is they're encouraging this NASH, people which can be used the non-radio labeled probes, radio label which is the P32, but non-radio labeled probes are which can be non-hazardous and which can be used very effectively and cheaper also comparatively this. So this is a method about how they can radio label random primers will be there, clinofragments, and the labeling will be there, radio labeled. But they label now the digoxin label, dig probes are coming, which is non-radio label, or biotin labeled probes are coming. You can use these one to detection of the what you can call as nucleic acids by targeting the specific one, and this will be highly effective, and it will be, you can use with efficiencies there. So the NASH is also very popularized by the WHO. So there are other methods which can be dot blot binding tissue plots, other northern blot methods are there, but these methods are the NASH is now the simple method which can be hybridized, putting on the membrane, and then you can use the autoradiography or color and chromogenic substances, you can use it, and then our fluorescent substances, you can study the detection of this genome and all. So recently, these techniques are very popular. Watch CRISPR technology. I think everybody knows, the faculty. CRISPR technology is one of the very important methods. People are exercising for detection of the viruses, the COVID, especially there are different methods are there in the CRISPR technology. The first one is what you can Sherlock method, where you can see the recombinase polymerase amplification, which convert the viral RNA to dsDNA. So here the T7 transcription generate complementary RNA will be there. So here this CRISPR technology, which is very popular now, which was extensively using, but Cas13 based one, and then this is a highly authenticated, and detection is very, very specific, and also you can see the low level viral RNA can also be detected, and because in the COVID, it was successful in the detection in this one. This is Sherlock assay, the COVID-19 S gene detection, COVID-19 ORF1 AB gene detection, one gene detection, because here the specificity is very high, and then you can see the detection of the COVID by using Sherlock assay, which is on the concentration level. So you can see in the especially in the x-axis, you can see that what level they can detection at the different genes. Now another one is what you can call as the second method, which is a bDNA method, which is endonuclease targeting CRISPR reporter will be here. That is what you detector method, assay method. So here the recombinase polymerase amplification will be there. This is what you can call RT-RPA is playing very important role. This is Cas12a. This what you can complex can be used here. Cas12a nuclease, which can be activated by target sequence, and the cleavage with the fluorescent RNA which is reported. These two methods. So the bDNA method, which is calling as the detector method, so endonuclease targeted detector method, and another one is the Sherlock method. Both are effectively using for detection of the viruses. So especially in the coronaviruses, they are popularized and also highly effective, and specificity will be there in this. And other methods also coming. I think the loop amplification method, these are the easy method and sensitivity very there. Here you can use the lamp. So this loop amplification, the primers are loop mediated here, and then they can amplify it. So this technology also very popularized now. So the loop mediated amplification lamp is very, very now widely using for the coronaviruses. So this the advantage of this one. So the loop mediated amplification simply required only the low which can low cost, especially instruments, and also water bath is enough. By visibly also you can see that. So this visible image just once you can amplify easy. The primary design is different than the normal PCR design. So it's simple and also very cheaper, and easily you can identify by visible, because by visibility the virus is the amplicon is present or not. So and also sensitivity is also equal to the PCR, but very cheaper and also very simple and very visible. You can detect. No need to go for what you can call as the quantification. This is only qualitative amplification method. So this is here also a lamp. There are different variants has come. There is a reverse transcription loop mediated isothermal RT lamp. So you can see the RT lamp also popular for this COVID, because this is a reverse transcription where you can see which can be used, and a sample mixed with reagent, and then RDNA loop PCR product will be there, and then you can easily identify visualization by easily. So then other techniques are coming here. This you can see the real-time PCR. Now everybody talking earlier. We are discussed about the qualitative PCRs. Now it is quantitative PCRs has coming by using different methods, advanced methods like a Trackman Assay method, molecular beacons method, cyber green dye method. So these methods are which are commercialized. So then you can give the sample automatically. They can make it out by using the what you can call as the instrumentation, and then they can give the result easily for the identification of the viruses. So the Trackman Assay is one of the where you can Trackman probes, which are very popular. We can identify the Trackman probes and amplify. You can do it, especially by using a forward and all. So the Trackman Assay is this. Here you can see the threshold will come, the cycle threshold will come, and the viral load can be automatically give the details how much threshold will be there. In the same way, molecular beacons also, which are another method which can be amplified and which can be by using different probes. In this one here also we can use fluorescent probes are there, and different probes we can identify. So the molecular beacons, they loop me here. You can see the stem and loop like that. So the other methods are the cyber green dye method. This is also very popular method which can detect the nucleic acids. So this here, the real-time Immuno Capture RT-PCR was used for detection of this COVID. So the cyber green dye which can be used. So this cyber green dye which can be amplify and then give the detection for the viruses. And now the very popular is the array technologies. Now this is the period of array technology after the human genome platform, which has evaluated. Now the human genome arrays are available. So in that time, so the revolutionized in the virology also. So the array technology has come. So this array technology, which can specifically on the specific target of the what you can call as identity. So there are microarrays and macroarrays are there. So these micro and macroarrays, which can be depending on the size sample, the macroarrays which can be large size sample can be used. That platform can use like a dot plot, but this was because due to the number of samples are limited and also volume will be high. But microarray is very popular now because microarray technology, these platforms already ready-made available in the market. So then you can make a detection of differential genes expression in the same pattern. You can see that this platform will be available in the volumes are very, very low. The micro slides and micro volumes will be there. RNA microarray is also available. So DNA micro also available. The RNA micro, which can use this for detection of the RNA viruses. So these platforms, the microarray platforms are very wide applications are not only viruses in the human genome, and also you can see the animal genome, where are the detections, mutations are there usually. In the same way, virus also you can use the RNA microarrays to detect the variation in this COVID, they popularized, and then it can become very, very unique features in the using this one. So this is a microarray platforms where the microarrays can be put on this platform, and then you can use the probes and labeled Psi3, Psi5 labeling will be there. And these platforms which can appear like different colors are there. So this the micro platform, which can express the both cells with RNA and different what you can call a microarrays we can use, express it in the cell. Suppose example RNA A, RNA B like that. So you can, depending on the color, so the arrays, so you can identify which is a positive, which is negative, like that you can array. This is ready-made platforms available. You can use directly for the detection. So this is what you can call as the viral my platform has microarrays available, very popular because it was the cDNA where different fluorescent labels which mixed with the microarray and coated with specific DNA probes. So this was already very popularized in the SARS period 2014. So it is commercialized for the what you can call reference platform will be there. So if you can take this platform and then you can compare with these viruses, either the different variants, usually you can detect which variant is there. So like that, the reference platforms are available now, and these platforms can be used to detect the variants on this. Other than this, so recently people says that we are sequencing and next generation sequences are there. So if you want to go for accuracy of the viral variants, so you can go for the next generation technologies like Solex, Solid, 454. These are different technologies which are next generation technologies are available. So which can be used for the ion Toronto and then AB's color space like that. So these next generation, so you can take the sample and either nasal or then swabs and then put in the medium, and then you can preloaded what you can call as the samples are available. So you can put it in the preloaded different what you can call as sequence, what you can detectors. So then you can plug it and then you can do it. So because ion Toronto, I worked with ion Toronto. So then you can put isolate the viral sample, collect the sample, put it in the vial and put it, and you can go for the sequence or nucleotide sequence analysis. Automatically it give within no time. You can see the sample within a very fast and high accuracy. So you can compare by using the phylogenetic analysis by using the tools, and then you can compare and how much homology is their sequence homology. Then you can say it which can be closely related, which domain, alpha, beta, gamma domain. So this is the sequence homology. They can study it by using the what you can call by using different signals and sequence different reagents. You can use ready-made reagents and then you can detect it. So this is the Solex flow. So you can see the Solex flow. You can put the sample. Automatically it can go on the flow which can run and give the sequence. And recently now the what you can call as the electronic people has come, and then the different other integration of the biosensor based diagnostics has come. So these biosensor based viral diagnostics are very popular because I have the collaboration with the NCRM, Tokyo, Japan, where there is a chemical engineering department is there in the Shibabara Institute of Technology and Tokyo University. I had the collaboration with that. So when we are developing what you can call as a chip-based viral diagnostics, because so this chip-based viral diagnostics, we can put the virus in the chip. So there is a small chip will design for specific virus, and then you can detect it within no time. And sensitivity is very high, cheaper, and also what you can call as there is no cross reactions in that. So this biosensor diagnostics is coming forward for the virus diagnostics. So this is the base here. The basic principle is what you can analyte will be there, sample and detection will be there. There you can see signal will be there and respond. You can detect. These are the biosensor based. I think we have written a book on the biosensors, especially biosensors of the different biological samples and all. So then, this is recently we had worked with these instruments, biosensor based, because this is the Raman based detection of the viruses. So this is enhanced because all of you might have known surface enhanced Raman spectroscopy, SCRC. Depending on the principle, what you had a collaboration with the what you can call Netherlands, and this kit has brought to India, and we had collected a lot of COVID samples, and then we did on free of cost, and we compared with the PCR. So this is very highly sensitive, and also this is a chip based one. So then is you can put a sample into chip, like a small cartridge will be there. So then you can link to this like a cell phone. So link to this automatically to detect it, and it can use the viral load compared with the control, and with the control you can keep it a healthy person one. So then it will give a reading by using the principle of the surface and enhanced Raman spectroscopy. So here this is another method has come, Optofluidic chip model. So here the Optofluidic chips are there, those the small chips which can load the sample here, and then this chip can be used to detect the virus very faster way you can detect it. So this is, you can see that how the they can method what you can see the faster. This is calling as Optofluidic chip model. So another one is this is calling surface plasma resonance, SPR. So the surface earlier I told the Raman, the surface plasma resonance, SPR biosensor rapid diagnostics. This is also we practiced I think this can be used for this detection of the viruses by using the plasma resonance surface SPR method. This is also fruitfully we worked and is working very well. And this is also in the SPR diagnostics are coming into the biosensor based viral diagnostic, especially in the occasion of this COVID. And another method we designed is the bacterial rhodopsin method. This is what we have designed with the collaboration. So with the different institutes, and we have isolated this bacterial rhodopsin. You know that bacterial rhodopsin has an optical property. So you can take this halobacterium and isolated the bacterial rhodopsin, and this rhodopsin we can put a slide and then coat it on a slide, and either you can coat antigen or antibody. If we can put into the what you can call a Raman Spectrophotometer, and then what happens here, the light will pass, the rhodopsin will be illuminated because the rhodopsin has the ability to light scattering will be there. So then the light, when the antigen binding, the light scattering is different. When they without antigen binding, the light scattering will be different. So you can compare. So either the control or the antigen binding, then you can use this technology. This is a new concept, which can be either you can coat the BRP with the antigen, or you can coat the BRP with the antibody, and then you can detect which one you want by using this Raman. So this way, so this is a nano diagnostic tool which can be used to detect the viruses by using latest methods. These methods we are involved and we develop and we are going to commercialize, and biosensors already are commercializing into the market with the help of the Tokyo, Japan people, and also bacterial rhodopsin also we are compared with the IASC, and then when also using the what you can call as these different institutions. So what we are going to see the collaboration, and this is also very cheaper. BRP is very cheaper, and also sensitivity is more for detection of these viruses. So with this one, I am highly thankful for the people who have given the opportunity to share a little knowledge what I know. So I think all of you know that how this gentleman has said. So this is very successful because the should not be where you can. So the virus, this is because called failure, because the diseases, everybody's scared about disease and virus, and that's why. So because the disease discomfort, which can be always will be there. So, but you have to be bear with us, and then so you have to learn a lot of things from the Omicron or the COVID, and this is a successful story of that. So with this one, I'm highly thankful the people has been opportunity. So with this small knowledge, I have given a talk to this. So thank you very much.
Thank you, sir. That was a wonderful talk from you. Thank you for introducing us to various new techniques in the field of viral diagnostics. Thank you so much, sir. To propose the vote of thanks, I would like to call upon Dr. D. Vasavidhatar ma'am, Telangana State President, Microbiology Society, India. Over to you, ma'am.
Appreciation.
So before vote of thanks, so please accept our appreciation certificate, sir.
Thank you. Please. Thank you very much for the certificate appreciation to given me. So thank you for the organizers. Yeah.
Thank you, sir.
Yes.
Hello.
Over to you, Vasavi ma'am.
Yeah, Mercy, before proposing the vote of thanks, I think we have to go to question and answer sessions if there are any.
Yeah, please.
Sir, good morning, sir.
Yeah, please. Good morning.
Sir, I'm Dr. Mansudan Reddy, sir. Gopaleti, sir, student.
Yeah, Chappan. Sir,
how are you, sir?
Yeah, fine. Master Bhavanara?
Sir, right. Sir.
Right, please.
Sir, one doubt, sir. During this SARS-CoV, there is a rumor, or I don't know whether it is true or false.
Right.
A few blood groups are less prone for SARS-CoV infection. Is it true or false, sir? They say O positive because they express low ACE receptors. That's why they are less prone for this infection. They said. Is it true, sir?
Yeah, you're right. I think what you said is that because the statistical analysis, they made analysis that which group has more prone in the same chikungunya as was happened in same earlier in the epidemic. So because people are taking a statistical, which group is positive, which group is negative. This is only statistical simulations only, but the scientific evidence has not it, because how these receptors of the blood group which can be prone for that like that. So still it needs more evidence should be required. But there are evidences that the majority of the people of the so-and-so group is more prone like that. This is a simulation only, analysis only. Scientific evidence is still required. Yes, you are right.
Thank you. Sir, another one, sir.
Please.
Like, long back, I have, like during the first phase of COVID, I had read an article in a newspaper saying that this coronavirus, though its intensity is more during the first phase.
Yes.
So any pathogen will not damage its host, but in the course of adjusting with the pathogen, with the host actually, it damages the host initially, but slowly it gets an understanding it with the host, and finally it will start living with the host, like how flu or even common cold viruses they do. And finally they say even this SARS-CoV also is going to be with a human being without affecting it in future. Is it true, sir?
Yeah, exactly. Because the reason is now you have seen the variants of the COVID. So from starting to now I think the Omicron, you can come and see that. So these Omicron, when enter into the body, the people who are suffering with more lung infections, TB, they are prone for the morbidity. So what happens? This virus is slowly, because changing their epitopes, and they are going to become a part of the, like you can see the earlier influenza, SARS, and so like that it can become like a natural phenomenon, and slowly it acquainted with our own genome, and then this will go into a normal phenomenon. So actually, I think the youngsters of the students should know that there is one recent phenomenon has come. All of you, I think, might have known, a defective interfering phenomenon, DI particle phenomenon. So the DI particle phenomenon says, that is Van Magna's hypothesis, viruses are not a pathogens. Viruses are they part of the human genome, and they escape, and then they can become like a pathogens. So that is a new phenomenon has come, that is a defective interfering particle phenomenon by Van Magnus, who proposed that whatever the viruses are there in the body, for example, so the DNA viruses, RNA viruses, so which can integrate with the human system, because if you can see the compare with the homology of the human genome sequence, so these retrogenes, retrotransposons exactly mimic like a human genome. So what he said, a part of the gene escape from the human, and then it will entangle and make itself replication, make it to their own pathogens. Pathogens is nothing but integration of the interference of that small gene set of the nucleotides with the human, and then become it to defective, that we are calling as disease. There is a new concept as coming slowly about what you said is the integration with the human genome, right?
So last one, sir.
Yeah.
Like in your slide, actually, I've seen till now, till Omicron.
Yeah.
The glycoprotein spike mutations were approximately 10 or 12 mutations, sir.
Right.
Whereas when it comes to Omicron, it is 32 mutations.
Yes, you are right.
So is it because of more mutations the virus has become less virulent, or is there any relation like that, sir, with that?
Yeah, it is more mutations will be there. What happens slowly, it will loses their virulency, and then slowly it becomes into what you can call as non-virulent. So then that's why you can get it herd immunity also. The body can slowly acquaint it, because the virulence you can compare with the first one, and now this one is not that much, but only you can get the slowly the fever, and then sometimes the body pains and all. Slowly it can go into the normal phenomenon, like a normal influenza like that. So it will go off, shortly it will go off.
Okay, sir. Thank you, sir. Thank you.
Thank you, Matsuda. Dr. Matsuda.
Sir, we received questions in the chat box, sir.
Yeah, please.
To protect ourselves from various COVID-19 variants, is it necessary to go for booster vaccination? This is the question, sir.
Yeah, only Covishield or Covaxin or earlier 2 doses depends. Please explain it. Yeah, this is true, because what happens here, you can see the vaccine platform. Covishield platform is different, and the Covaxin platform is different. So Covaxin platform is a kill vaccine, because they are using Covaxin is a whole virus killed virus, but Covishield is a recombinant virus. Generally, the scientific mean says that Covaxin, which is a killed form, you need a booster dose so it can enhance the immunity. But Covishield is a recombinant form. Generally, what the scientific people says, so Covishield is a recombinant form like hepatitis B, HBS vaccine. So it will be sustained in the body. It will go enhancing continuously. So that's why the people more, the Covaxin, you have to take booster. In general, Covishield is not required, but still the people says put the Covishield also booster like that. You know that sometimes the Covaxin people are taking the Covishield booster, Covishield people are the Covaxin, but that is not correct. Covishield is recombinant, but the booster is generally not required according to scientific reports. But the Covaxin is required because it is a killed vaccine, so you required the booster for that. That is true. Right.
So,
yeah, some Madhuri is asking that
Yes,
patient
sir.
developing diabetic irrespective of their age. Because the diabetic, because there are different diabetics are there, but due to the COVID, what happens? So the immune system, which can be, because COVID-19 recovered patients developing diabetic irrespective of the age. That's true. Because what happens, the COVID when comes to now, the immune system, some of the genes, they are in the COVID, that the genes which can immune, now recently you have seen in the Omicron, the immune suppression genes also there. So these genes which can go and suppress the immune system, so the immunity also will going on changing in the system. So that's why some people, they are suffering with the diabetes, some people suffering with the blood clots, some people suffering with the lungs infection, like that. These are calling as morbidity. Secondary infections are coming and super infections are also coming. There are some immunological disorders are coming due to the virus integration with the system of the normal mechanism. That is true, because there are different type of some people are suffering with diabetes, some people are suffering with the kidney problems, some people are suffering with the lungs, and some people are suffering with the clots or like that. So there are a lot of the morbidities are coming in the system. Yes, you are right.
Sir, I'm sorry, sir.
Yeah, please.
Sir, is it that because in COVID multiple organs are involved? Suppose if not all the patients are getting this diabetic as a complicated what do you call it, case, is that because if this virus involves a spleen actually, where production of this insulin will be affected, because of that they may be getting diabetes also, no, sir?
Yeah. There are reasons. Their reports are coming like that. You are right. Yes.
Thank you.
Yeah, and also some people. TB patient, now the Omicron is affecting more on TB patients. So because TB prone, already TB is there with the patient. So the Omicron went there, it can aggravate. So that's why people say, so people are suffering with TB, first we can take the treatment that, and then we can, so we will be safe, like that they're telling. In the same way, other patients also there. Some of the patients who are suffering with the lung infections also, some lung nodules, TB patients, and some of the spleen, some of the patients who are suffering with the liver. Suppose hepatitis B patient is there infecting with the HBV. When the COVID has come, so the COVID also can make a role on the HBV, and those people, what happens, the infection will be more aggravated, and there is a prone for the morbidity will be more in those patients. They're like that. This is calling as super infections are there, co-infections are there, and then secondary infections are there. So the morbidity is going on, changing the human system. Yes.
Thank you, sir.
Sir,
Yeah.
thank you very much, sir, for
Thank
your
you.
valuable time for us. We honored with your excellent presentation, sir. Thank you very much.
Thank you, Thank you, Ms. Antipri. Thank
Over
you.
to Vasavi, ma'am.
yeah, thank you. Thank you, everyone. Good morning, all the dignitaries and delegates. It's my pleasure to propose vote of thanks for this technical session. Professor Sai Gopal, sir, has enlightened us.
Am I audible?
Yes, ma'am.
Yes, ma'am. Carry on.
Okay. Professor Sai Gopal sir has enlightened all of us on recent trends in diagnosis of Omicron with emphasis on various things. Sir, you told that you have a very little knowledge, but that's not true. Actually, you have showered us with a lot of information. You also have given the emphasis on possibility of plant viruses infecting humans and the mutations in the genes coding for the spike proteins, variants of coronavirus and their designations, phylogenetic relationships between coronaviruses, and even a single mutation can cause a structural variation. Then study of variants by molecular analysis and variation of the symptoms accordingly. It is interesting to note that the methods of diagnosis also should be upgraded as the virus goes on changing its structure. Professor Sai Gopal has explained the different diagnostic methods of coronaviruses, like older and new methods, sir. Both you have stressed very well, like the rapid diagnostic test, nucleic acid amplification test, and the knowledge of endo period, that is crucial. It is important to differentiate between symptomatic and asymptomatic patients, and also false negative tests. So among the diagnostic methods, sir has stressed on older methods like fluorescent antibody technique, radioimmunoassay, ELISA, and the newer methods like luminescent assay, dot blot, immunoblotting, and the lateral flow diagnostic methods. So as sir said, the serological tests have some disadvantages. So newer methods, advanced methods are being used, like PCR, Immuno Capture Pcr, loop amplification, real-time microarrays, and chip-based techniques. This is the newer things we have known today, and the use of bacterial rhodopsin. So the very sensitive and economical tests you have narrated, sir. You have beautifully presented the various methods in an understandable way. And also you have answered the questions regarding the comorbid situation, sir. Thank you very much, sir, for making us aware of such vast information on diagnosis of Omicron and other variants of the coronavirus. And please, can you show that book or give a link of the book which you have written, sir? I hope so that we can get much more information.
Thank you.
Thank you very much for giving me this opportunity to the organizers.
Thank you very much, Dr. Vasu.
Sir, I request you please accept our appreciation certificate, sir.
Thank you, Amma. Thank you, Santa.
Thank you so much, sir, for your valuable time for us.
Thank you, Amma. Thank you,
sir.
Honor. Thank you very much.
We blessed very much, sir.
Thank you. I love you. Thank you very much. Sir. Thanks.
Thank you, sir. I hope every one of us have thoroughly enjoyed the session. Let's move on to the second speaker of the day, Dr. Rukhsana Rahan, ma'am, Associate Professor, Department of Microbiology, US Bangla Medical College, Bangladesh. To introduce the speaker, I would like to call upon Dr. S. Mrudula, ma'am, Assistant Professor, Department of Microbiology, Palamuru University. Over to you, ma'am.
Okay, thank you. Good morning to all. Thank you for giving me the opportunity. Now I take privilege of introducing the second speaker of this session, Dr. Rukhsana Rahan. Let me give a brief introduction of Dr. Rukhsana Rahan. First, I would like to focus on her educational qualifications. Dr. Rahan has done her MBBS from Medical College for Women and Hospital, Dhaka University, Dhaka, Bangladesh in 2000. She obtained her MPhil in virology from Bangla Bandhu Sheikh Mujib Medical University, Dhaka, Bangladesh in 2010, and received PhD in medicine from University of Malaya, Kuala Lumpur, Malaysia. Regarding her work experience, Dr. Rahan has work experience of 20 years in the field of medicine. During these 20 years, Dr. Rahan has worked as lecturer, medical officer, director of pediatric hospital, and assistant professor in Bangladesh from 2001 to 13, and as lecturer in the Department of Microbiology, Faculty of Medicine, AIMST University, Kedah, Malaysia. Currently, she is an associate professor, Department of Microbiology, US Bangla Medical College, Dhaka, Bangladesh. Dr. Rahan is the head of US Bangla Molecular Lab. Doctor is also a senior consultant to American Society for Microbiology USA, and she has been serving as director at Center for Medical Research and Training since January 2021. Dr. Rahan teaches immunology, microbiology, and virology for both undergraduate and postgraduate students, and also involves in research guidance. To her credit, she has published 18 research articles in various reputed journals and also presented her research work in various conferences across the world. Dr. Rahan also serves as a member for various committees across the world. Let me mention few of them. Dr. Rahan is General Member of Bangladesh Medical and Dental Council, Indian National Association for Study of the Liver, and American Society for Microbiology. Founder member and EC committee member of Society for Medical Virologists Bangladesh. Dr. Rahan is also an editorial board member of the Journal of the Academy of Clinical Microbiologists, member of steering committee of Hepatitis-Free Pahang Association. Dr. Rahan is a reviewer for 2 journals, namely Clinical and Experimental Hepatology and Medical Journal of Malaysia. And she has many more achievements to her credit. Now, without any delay from my side, I shall hand over the proceedings to Dr. Rukhsana Rahan to enlighten us with her talk on SARS-CoV-2 variants and vaccines. We are very happy to have you here, ma'am. Please welcome. Thank you.
Thank you very much, and a very good morning to you all. I am very honored, and I'm really very thankful to the Department of Microbiology of Palomar University for inviting me in this platform with such eminent professor to speak about coronaviruses. I really don't know how much I can justify, and especially hearing after, especially once I heard Professor Sai Gopal sir, I'm really very impressed. And in fact, I have learned by myself a lot regarding this Omicron diagnosis letters. Without further delaying, I would like to start my talk on SARS-CoV-2 variants and vaccines. So sorry, let me fix it. So regarding SARS coronavirus, we already know that this belongs to a subfamily of coronaviridae, a family of coronaviridae, and order Nidoviridae. So there are namely 4 SARS coronavirus, alpha, beta, 4 genera, alpha, beta, delta, and gamma. And our SARS coronavirus 2, the topic of interest, belongs to beta group. The SARS coronavirus structure, if we look a little bit, then they are spherical to pleomorphic particle, meaning measuring between 80 to 160 nanometer in length. And it contains 4 structural protein, envelope spike membrane, and nucleocapsid. So these protein together form the spike membrane, and envelope protein together form the envelope of the virus. The M protein is the most abundant, most responsible for the shape of the envelope. The E protein is the smallest structural protein. N protein remain associated with the RNA forming a nucleocapsid inside the envelope. Polymers of S proteins remain embedded in the envelope, giving it a crown and shaped appearance that gives the name coronavirus, which actually means the crown. This is actually an electron micrograph of heavily infected cell with coronavirus, SARS-CoV-2. So that is how this apoptotic cell looks like under electron microscope. The major target for the neutralizing antibody or vaccine development is the S protein. S protein has got several other importance for SARS-CoV-2. It has got 2 subunits, so S1 and S2. S1 subunit contains a signal peptide followed by an N-terminal domain and receptor binding domain. S2 subunit contains conserved fusion peptides, haptid repeat 1 and 2, a transmembrane domain, and a cytoplasmic domain. S1 binds to ACE2 receptor of the host cell, and S2 promotes membrane fusion. And another transmembrane protease serine 2, which is also known as TMPRESS2 purine, is also called S protein cleavage enzyme, mediate the cleavage of the S1 and S2 site in infected cells, promotes subsequent TMPRESS dependent entry into the target cell. If we look at the genome a little bit, SARS-CoV-2, the reference gene of the SARS-CoV-2 genome is 2900 and it's around 30,000 nucleotide in length, comprises of order of 5' replicase, ORF1ab, SEM, N, and 3'. The predicted replicase ORF1ab gene is 21,291 nucleotide in length. The ORF1ab polyprotein is predicted to be cleaved into 16 non-structural protein. ORF1ab is followed by a number of downstream open reading frames. These includes spike, ORF3a, envelope membrane, and nucleocapsid gene of length 3,822, 828, 228, 669, and 1,260 nucleotides respectively. ORF8 gene located between M and N gene, and finally the 3' and 5' terminal have length of 265 nucleotide and 229 nucleotide respectively. So regarding the variants, we know that the virus replicates, it makes copies of itself. It is normal for small changes or mutation to occur. A virus with one or more mutation is then considered as variants of the original. When a virus circulates widely throughout a population, the chances of its mutating becomes higher, and considering RNA virus, it is even more higher. So while most variants have little impact on the properties of the virus and the impact it has on the community, some can have larger impact, as we can see with the ongoing pandemic of the SARS-CoV-2. So why variant surveillance is important? Some of the potential consequences of emerging variants are the following: ability to spread more quickly in people, ability to cause either milder or more severe diseases in people, ability to evade detection by specific viral diagnostic tests, and decreases susceptibility to therapeutic agents such as monoclonal antibody. It can also modulate the ability to evade natural or vaccine-induced immunity. So by the time we see different nomenclature of this SARS-CoV-2, the established nomenclature system for naming and tracking SARS-CoV-2 genetic lineages by GISID, NextStrain, and PanGo are currently and will remain in use by the scientists and in scientific research, who and its international network of experts developed a variant classification scheme that defines 3 classes of SARS-CoV-2 variants: variants of interest, variants of concern, and variants of high consequence, which was previously there, but it has been changed now. The World Health Organization announced that SARS-CoV-2 variants of interest and variants of concern will be named based on the Greek alphabet for the purpose of the public use. So what is variant of interest? With genetic changes that are predicted or known to affect virus characteristics such as transmissibility, disease severity, immune scape, diagnostic or therapeutic scape, and identified to cause significant community transmission or multiple COVID-19 clusters in multiple countries, which increasing relative prevalence alongside increasing number of cases over time or other apparent epidemiological impacts to suggest an emerging risk to global public health are considered as variant of interest. Variant of concern, SARS-CoV-2 variants with genetic changes that are suspected to affect virus characteristics with some indication that it may pose a future risk, but evidence of phenotypic and epidemiological impact is currently unclear, requiring enhanced monitoring and repeat assessment pending new evidences. These are variants under monitoring. So whatever the new variants are coming, basically are put into this variants under monitoring. And variant of interest. Sorry, the variant of concern are the variants. That means definition of variant of interest, and through a comparative assessment has been demonstrated to be associated with one or more of the following changes at the degree of the global public health significance. Either it may increase the transmissibility or detrimental changes COVID-19 epidemiology, or increase in virulence, or change in clinical disease presentation, or decrease in effectiveness of public health and social measures, or available diagnostics, vaccine, and therapeutics. So all the variants of interest, after careful consideration when it poses any of these threats, can be considered as variant of concern. So already said about the variant of monitoring, which basically all the variants come across, we monitor and then goes for variant of interest or then variant of concern. So a previously designated variant of interest or variant of concern, which has conclusively demonstrated no longer or major added risk to global public health compared to other circulating SARS-CoV-2 variants, can be reclassified. So currently there are two variants of interest, lambda and mu. So the lambda one is first identified in 14 June 2021, and mu was first identified in 30 August 2021 in Colombia. So basically these two are circulating for quite long, but still they are not much concerned as Omicron or Delta. So that's why they are not transferred to variant of concern yet. So right this moment, there are 5 variants of concern: alpha, beta, gamma, delta, and Omicron. So previously we have seen alpha, beta, gamma, delta to cause its devastating impact on throughout the world. And still Delta is not over. Delta is there, but the current global situation, I mean, the surge is on Omicron we are battling with. So alpha, there are in Pango lineage, it also is known as B117, and GISID, it's also called GRY, and next enclave 20I. So there are different entity for classifying them. First identified in United Kingdom in September 2020. Beta was first identified in South Africa in May 2020, and it is also known as B1.351. Gamma is P1 in Pango lineage, and it was first identified in Brazil in November 2020. And Delta, the one identified in October 2020, and the second devastating second wave we have seen basically is due to the Delta, is also known as B16172. So currently the circulating, the main concern is Omicron, which is also known as B11529. And it was first identified in South Africa in 24 November 2021. It's not long, it's just 2 months. So this is the original strain of, SARS-CoV-2, and these are the different strains. And here we can see their different changes, different positional changes and mutation. This is the N-terminal domain of the S1, and this is the receptor binding domain. This is the cleavage protein, fusion peptidase, haplotype. So we can see most of the mutations are found in the S1 part of the spike protein. So that causes the changes in all these variants and their impacts. If we go a little bit details on this, then alpha, you can see there are many mutations, 23 mutations in fact, And importantly, there was N501Y mutation, which increased the vital binding affinity with angiotensin converting enzyme 2 receptor. H69 deletion and V70 deletion mutation, which was potentially associated with immune evasion and affected S gene polymerase chain reaction assay. That means the PCR assay resulting in S gene target failure. So before we, many of us who have worked in lab side, we have seen that the kits, PCR kits using 3 gene and especially using S gene, there were many S gene missed. It mostly, we got it in 2020 and the first part of the 2021 before the Delta. So it was one of the measure to identify indirectly that this can be caused by the alpha variant. So there is also important P681H mutation, which potentially facilitated the epithelial cell entry. So the beta is also known as B1351, was discovered in Brazil. So 3 mutations of the receptor binding domain in the spike glycoprotein of the virus, N501Y, a change from asparagine to tyrosine in amino acid position 501, and K417N and E484K. 2 of these mutations, 484 and 501 positions, are within the receptor binding motif of the receptor binding domain. This receptor binding domain is one of the major target for the neutralizing antibody. So 2 mutations found with the beta variant 484K and 417 are not found in alpha variant. Also, beta variant does not have a 6970 deletion mutation found in the other variants. 484 amino acid change, a receptor domain mutation, was reported to be associated with the escape from neutralizing antibodies, which could adversely affect the efficacy of spike protein dependent COVID vaccines. So vaccine efficacy was altered by these mutations for the beta variants. The gamma variant has mutation that help it evade antibodies from previous infections or from vaccination, and thus may be able to more easily reinfect people who have already had COVID-19. 17 amino acid substitution and 10 of which is in its spike protein, including these 3 designated to be of particular concern, 501, 484, and 417, which we have seen in beta variant as well. The gamma variant has a number of mutations compared with the original. It's also 501 is also present in alpha. This mutation allows the virus spike protein to bind more easily to human cell, which could make it more infectious. And here comes the delta. It has mutation in the gene encoding the SARS-CoV-2 spike protein, causing the substitution of 478, 618, L52, which are known to affect transmissibility of the virus, as well as whether it can be neutralized by antibodies for previously circulating variants of the COVID-19 virus. It is thought to be one of the most transmissible respiratory viruses known, whereas if the original virus strain could affect from one person, it could transmit to 2.4 to 2.6 person. The alpha could have transmitted 4 to 5%, but in case of delta, we got it like 5 to 8%. So till the discovery of the Omicron, the Delta was known as the most transmissible respiratory virus ever known. The viral load of the Delta infection was 1,000 times higher than those in infections caused by other variants. So that also was very much prominent in the diagnostic sectors, like while doing PCR, we have seen the viral loads, the CT values were much less in case of Delta infections. And the current one, the Omicron variant, which is also known as B11529, was first reported from South Africa on 24 November in 2021. 26 November, a technical advisory group declared it the name Omicron. 32 mutation affect the spike protein, the main antigenic target of the antibodies generated by infections and many vaccines widely administered. Many of those mutations had not been observed in other strains. The variant is characterized by 32 amino acid changes, 3 small deletions, and 1 small insertion in the spike protein compared with the original viruses, of which 15 are located in the receptor binding domain. The standard, it has got two other sublineages, namely B2, B3. B1 and BA1 and BA2 share 32 mutations, but they differ by 28. That is why they are two different entities. This makes them as different as some other major variants, and it has been suggested that BA2 should receive its own name based on the Greek alphabet, but still not done yet. Omicron multiplies around 70 times faster than the Delta variant in the bronchi, but evidence suggests that it is less severe than the previous strains, especially compared to the Delta variants. So Omicron might not be able to penetrate deep lung tissue. That is one of the major reasons why it is less severe. Omicron infections are 91% less fatal than the Delta variant, with 51% less risk of hospitalization. Early figures suggest that double vaccination offers 30 to 40% protection against infection and then around 70% protection against hospitalization. A recent third vaccine doses boosted effectiveness against infection to around 75% to 82% for severe diseases. By comparing the variant structure with that of the original version of SARS-CoV-2, they have begun to shed light on which features of the highly mutated virus have enabled it to evade the body's immune defenses while also maintaining its ability to attack a person's cell. And they have begun to unpick why Omicron seems to cause milder diseases than the previous variants. So already said there are more than 30 mutations in the spike protein, and it has been observed that 15 of the Omicron spike protein mutation found in the receptor binding domain region of where the ACE2 on the person's cells to gain entry. These changes, along with 11 mutation in a region of the spike called N-terminal domain, have completely remodeled the areas of the protein that are recognized by the neutralizing antibodies generated after the vaccine against SARS-CoV-2 or with the natural infection. With such a big shift in shape, there's a huge question over how Omicron can still bind strongly with ACE2. Still, with these major changes, they have adopted a very elegant molecular solution where the mutations are mediating immune evasions while enhancing receptor binding. Although these mutations in RBD hinder its ability to bind ACE2, other mutations strengthen to bind it. So why is Omicron seems to cause milder diseases than other variants? There's a big question. So there are some probable hypotheses on that. So several researchers have mentioned. So one of that, it seems that there are more difficult infection. They have difficulties to infect lungs than nose and throat. So there are two mechanisms, proposed mechanisms. The first one involves an enzyme on host cell called TMPRESS. As I said, this is a enzyme which cleaves the spike protein into S1 and S2 and expose a region that embed into the cell's membrane. Eventually, the virus fuses with the cells and injects its genetic material directly into them. And there is another way of entry of SARS-CoV-2 that is a slower pathway, involves the virus entering host cell through bubbles known as endosomes before releasing its contents. So several groups have found that Omicron prefers the slower route, that is the slower route, that is that bubble, the forming the endosome before releasing its contents inside the cells, not the other one, the TMPRESS induced split of the S1 and S2 region and then to fuse directly inside the cell. Interestingly, the Omicron variant is less efficient for this TMPRESS pathway, one of the reason is the higher level of temperature in the lungs than in the upper airway. So that is probably why these Omicron variants prefer to infect for nose and throat, not in the deep inside the lung. Some group have reported evidence for a slightly different mechanism, that either of the other two, like I said, the slower method or the direct entry method, they suggest instead that Omicron mildness is related to the ACE2 receptor. To bind to ACE2, the virus's receptor binding domain needs to flip from a down to an up position. In a preprint, Chan and his teams have reported evidence that Omicron's receptor binding domain has difficulty moving into the up conformation because of a structural change induced by one of its many mutations. So as a result, Omicron requires higher level of ACE2 to fuse with host cell than do other variants, and lung cells generally have much lower ACE2 levels compared to the cells in the upper respiratory tract. So because inside lung has less ACE2 levels, so Omicron cannot enter inside as much as it can enter inside the cell of the nose and throat or the upper respiratory tract. So there are still a lot many to know about Omicron. In fact, a lot many still we require to know about SARS-CoV-2 itself. So this is the three-dimensional pictures of the spike protein, because the spike proteins are quite important in every aspect of SARS-CoV-2 infections. This is the regular one. This is the S1 domain. This is also the S1 domain, and this is the S2 domain. For better visualization, we can okay, never mind, the S1 chain. So this is the S1 chain, and this is the antibody binding domain, that is the N-terminal domain. This is the receptor binding domain centrally, and the main domain targeted the neutralizing antibodies, you can see here. This is the S2 chain, and here is the haploid repeat 1 fusion helix, and these are the RGD motif. The arginine glycine aspartate is commonly used by viruses to bind cell surface integrins. These are the different variants, and the red highlights are the mutation sites. So in general, at a glance, what we can see that Omicron has got the maximum mutations in its spike protein than any other variants of concerns before we talked about. Due to all these mutations, there are some significant changes in the patients of the Omicron. Earlier, we have seen dry cough, fever, chills, tiredness, muscle pain, diarrhea, sore throat, shortness of breath, chest pain, runny nose, conjunctivitis, loss of taste, a rash on skin as the common symptoms for COVID-19. But nowadays, predominantly what we can see, runny nose, headache, fatigue, sneezing, sore throat. And for the Delta one, what we have seen, fatigue, headache, anosmia, sore throat, cough, more prominent. So nowadays, Omicron is predominantly as same as the common cold. It is also termed as Omiflu, but still it's not common cold because common cold doesn't cause any death of the patient, but Omicron has that potentiality to do. Omicron can cause reinfection. It is 5.4 times greater than that of the Delta virus. I mean, the chances of reinfection is 5.4 times greater than that of the Delta variant. This implies that the protection against reinfection by Omicron afforded by the past infection may be as low as 19%. So current situation of Omicron worldwide. So what we can see from these statistics, we can see in 2020, this time, and 2021, and this is the January 2022. And you can see the graph, how rapidly Omicron is transmitting. It is already said that 70% more transmissible than Delta variant. So the sharp rise of Omicron variants. Here, this is the Delta rise, and this is the Omicron rise of the cases throughout the world. But interestingly, as we speak about that, the Omicron variant probably is less severe than the other variants. So this graph shows that the January 2021, during the peak time, the death rate was much higher than this Omicron time, and where the inflation of the cases are much higher. What we can see, this is the South Africa, where these variants were first detected. And what we can see that during the Delta time period, this is the highest peak, and the Omicron wave was much higher. But the deaths and the hospitalizations are much, much lower than what we observed during the Delta wave. So here also the similar graphics of, showing the ICU admission is much less than Delta wave. The oxygen requirement is much less than the Delta wave. So in altogether, Omicron is much more contagious, definitely, but because of its less severity, we are in better situation than the previous time. So talking about the vaccines of the SARS-CoV-2 is COVID-19 vaccine. The most of the platform of vaccine candidates in clinical trials are focused on the coronavirus spike protein and its variants. So platform involve nucleic acid technologies, non-replicating viral vectors, peptides, recombinant proteins, live attenuated virus, and inactivated viruses till now. As of 12 January 22, 9 vaccines have got WHO emergency use listing procedures. These are the Pfizer, AstraZeneca, then Johnson & Johnson, Moderna, Sinopharm, Sinovac, then Covaxin, Covax, and Novavax. So Pfizer, BioNTech, Moderna, they use the messenger RNA method from the inactivated one Covaxin. CoronaVac, they all use inactivated method, viral vector vaccines. Among viral vector vaccine, most importantly, Oxford AstraZeneca and Sputnik V. And subunit vaccines, none has been much well known till now. So there are many different differences in the dose and the temperature and all, but the most similar thing is almost all the vaccines, they are using, they are targeting the spike proteins. Only the Sinovac, one from China, they have targeted the whole virus, whole inactivated viruses. Otherwise, all others use spike proteins. So the messenger RNA vaccines have a strand of genetic material called RNA inside a spatial coating. That coating protects the messenger RNA from enzymes in the body that would otherwise break it down. It also helps the messenger RNA enter the dendritic cells and macrophages in the lymph node near the vaccination site. They instruct for the cells on how to make a piece of spike protein that is unique to SARS-CoV-2. Since only part of the protein is made, it does not do any harm to the person vaccinated, but it is antigenic, and once synthesized immune reaction starts against it. After the piece of spike protein is made, the cell breaks down to messenger RNA strand. It is important to note that the messenger RNA strand never enters the cell's nucleus or affect genetic materials. That is one advantage for messenger RNA vaccine, and this is the first of this kind of vaccines ever using for human populations. Inactivated vaccines, we already know, is a vaccine consisting of virus, particle, bacteria, or other pathogens that have been grown in culture and then are killed to lose disease-producing capability. No chance that they can revert back to their virulence. This is their advantages, and can be given to anyone in any time. And disadvantage, they make weaker immune responses and multiple doses are required. Viral vector vaccine. A live vector vaccine is a vaccine that uses a chemically weakened virus to transport pieces of the pathogen in order to stimulate an immune response. So adenovirus are mostly used. The Oxford AstraZeneca vaccine is a vector vaccine where modified chimpanzee adenoviruses has been used as vector, which cannot replicate, acts as vector to transfer the SARS-CoV-2 spike protein. The adenovirus is called replication deficient because some of its essential genes were deleted and replaced by a gene coding for the spike protein, and that made him a better vector for such kind of vaccines. Following vaccination, the vector enters the cells and releases its genes, which goes to the cell nucleus, transcribe into messenger RNA, and the translation and translate into proteins elicits immune responses. Subunit vaccines use only part of the target pathogen to provoke a response from the immune system. This may be done by isolating a specific protein from a pathogen and presenting it as an antigen on its own. So these are the targeted we can see for the SARS-CoV-2 vaccines used. So the major target used for the SARS-CoV-2 is the S protein. So it consists a membrane distal S1 subunit and membrane proximal S2 subunit and exists in the virus envelope as a homotrimer. The S1 subunit determines receptor recognition via its receptor binding domain, whereas the S2 subunit is responsible for the membrane prefusion, already said, which requires the virus to enter inside the cells. The previous coronavirus like MERS-CoV and the SARS-CoV-1, they all, and along with this is now on the SARS-CoV-2, the receptor binding domain is located in the C-terminal domain of the S1 subunit, but in some coronaviruses, the N-terminal domain of the S1 subunit can be used for receptor binding, or might also be involved in the virus attachment of the host cell by recognizing specific sugar molecules. The S2 subunit contains the fusion peptide connecting region and haptid repeats and 1 and 2 around a central helix as a helix-turn helix structure. So structural evidence has proposed a model for the rearrangement of SARS-CoV-2 S protein following recognition of the host cell receptor. So S protein can be proteolytically cleaved at both S1 and S2 and S2 cleavage side, releasing the structural constraints on the fusion protein. The engagement of the receptor binding domain with its cellular receptor, SE2 receptor leads to the dissociation of the S1 subunit and initiates the refolding of the process of the S2 subunit, which protrudes the fusion protein actually at its end for membrane fusion. So, but basically what I want to say that receptor binding domain is the outer part, then the this fusion protein or the S2 protein. So that is why this receptor binding domain has got some advantages to be used as the major candidate or the target for these vaccines. The surface, the surface of the S protein is extensively shielded from the antibody recognition by glycan, and with the exception of the receptor binding protein. That is why, again, this is easy accessible than any other domain of the S1 or S2 part of the spike protein. Most of the SARS-CoV-2 neutralizing antibody binds to the receptor binding domain and block receptor binding domain and the ACE2 receptor interaction, inhibiting the virus attachment. This receptor binding domain is an attractive vaccine target also because it elicits high quality relevant antibodies while avoiding the potential risk of any kind of adverse effects. And this also contains epitope for the T cell responses as T cell responses. Also, this is also proved in other coronaviruses like MERS and SARS-CoV-1. So these RBD-based antigen have been described in, many other studies for the best candidate for the vaccine development. And now, like the N-terminal domain and the S2 subunit, if I talk about the N-terminal domain, also contains epitope for SARS-CoV-2 neutralizing antibody. This N-terminal domain targeting neutralizing antibody generally do not directly block receptor binding, but rather interfere with the receptor binding. So they interfere or restrain the S protein conformational changes required for the pre-fusion to the post-fusion transition. It is also notable that the SARS-CoV-2 N-terminal domain targeting natural antibody generally exhibits lower neutralizing potency than this receptor binding domain specific antibody. So and also these N-terminal domain can exhibit T cell responses. So and furthermore, this N-terminal domain reduces lung abnormalities. Previous studies showed that in mouse model, the immunogenicity and protective efficacy of the N-terminal domain proteins were weaker than the RBD protein. So that is why, again, this domain is much less important than receptor binding domain till now. Yet so far, these vaccines, probably all these reasons that N-terminal domain vaccine target has not been reported till now for SARS-CoV-2. So the S2 subunit is targeted also for, as I said, the main target is S1. The S2 is much inside, so that made it a bit difficult to get the access for neutralizing antibody. And also many studies shows that S2 subunit targeting antibodies isolated from the convalescent patients showed weaker neutralizing activity against SARS-CoV-2 than receptor binding domain antibodies. So there are other proteins like membrane, envelope, and N protein. So unlike the S protein, coronaviruses, M protein, and E proteins are poorly immunogenic for humoral response. And that is why they were, they were never been used for or targeted for the vaccine development. The N protein is the most abundant of viral protein, and it is highly immunogenic during coronavirus infection. It is a major target for antibody response and also contains T cell epitopes. N-specific antibodies were reported to protect mice against mouse hepatitis viruses and different other coronaviruses. But NT and immune sera could not protect SARS-CoV-2 infection from getting enter inside the host cell. However, its potential for coronavirus vaccine target was largely undermined as because of all these. But there is still now no such vaccines were targeted for ME or N protein. So already we know the whole virus targets and a whole virus or inactivated virus or vector virus. We know a lot about this by the time and their advantages and disadvantages because these are all old methods, but mostly they all targeted S protein and especially the S1 region. So how effective of these virus, these vaccines till now? So here we can see the original variant the AstraZeneca, Johnson, Pfizer, Moderna, they are quite effective in the original virus, but less effective in Delta, Beta and Delta, if I compare with the original and the Alpha one. And so far, this Omicron, still there is no graphical representation of this, but this Omicron also had caused less effectiveness for all these vaccines with only 1 or 2 doses. So this is the 2-dose completed people showing the much higher immune responses against SARS-CoV-2 different variants. So it's not the first dose, the second dose for especially Pfizer, Moderna, and AstraZeneca showing higher responses. So there's a question like even after vaccination, many people are getting infected and many are dying. So then what is the necessity for giving vaccinations? So still, because of this continuous changing, it's been very difficult for controlling or development of all variant kind of vaccine yet now. But whatever the type of vaccines or existing vaccines, they can give some kind of protection against this death or hospitalization. So here we can see unvaccinated people, the death rate among unvaccinated people are much higher than the vaccinated people. So before vaccination and after vaccination also, there is a lot of changes. This is the death rate. The lower one is the death rate of vaccinated people, and this is the death rate among the unvaccinated. For Alpha, Beta, Delta, there is almost 100%, 90 to 100% decrease of death rate than the unvaccinated people. So there's again question, is the available vaccines effective against Omicron? So not 100%, because we already have seen there is a huge major changes in the structure of the S protein, which is the major target for most of the vaccines. So they don't give much protections, especially after first and second dose, but the booster dose can protect from the severe diseases around 80 to 85%. And it's 90 and more than 90% protection against death, at least 60 days after being administered. So that gives the importance of getting the booster doses. There are other parts of the immune system, such as T cells, that can fight also the COVID-2, as we, from previous discussion, we saw that S1 domain can exhibit some T cell responses as well. So, and also natural infection. Once we get natural infection, we get immunity against all sorts of antibodies, against all kind of antigens. So that also gives some cross protection against any kind of variants. So altogether, this helps to give some sort of protections. So there is a raising questions, the Omicron a blessing in disguise or a curse? So as on now, the preliminary reports on Omicron, the new variants suggest that this variant is highly infectious but causes mild illness. Though it is premature to conclude still, but if Omicron is truly infect many people and causes mild disease in them, it may be the beginning of the end of this deadly pandemic. So already we have seen in South Africa, within a month, the peak of the Omicron came down, and there was none died from Omicron. Still now, the death mostly worldwide we are watching are due to the Delta variant, and there are few for Omicron. And for Omicron, those who died mostly died because they were not vaccinated and they were overburdened with so many other comorbidities. So if these highly contagious variants are able to infect most of us worldwide, then there will be a natural protection top of that vaccinations, and which will help at least all of us to get to gain some kind of immunity from any kind of variants we coming to, generating next. So in a way, this can be blessings for all of us. So this variance prevention is all like before. So on top of that, we need to get the booster doses and future focus, global constant surveillance to identify potential variants, develop modified vaccines, including the virus-escape mutants, and especially in combination of different proteins. So like now they are targeting only one antigen, one target. So it can be better if we can produce a vaccine targeting many other antigens. So enhance production of vaccines globally, enhance vaccine program worldwide, vaccination of animal hosts to prevent development of mutation variants in them, as it is said that Omicron variants, Delta went to mice, and then there is this transformation, and they gain these different mutations and this immune evade mechanism, and then transfer to human. So we need to look after for all these animal hosts. And as some other raised the question whether or not we can get it from plants, or so yeah, there is a high chance. So we need to keep all these surveillances on. And the target therapy against important replicating enzymes can help to fight back against COVID. With that, I thank you all. Thank you very much.
Thank you, ma'am. Thank you for an amazing session. It was a detailed talk about various variants of coronavirus. And you have also explained about various vaccines which are coming out now. Thank you so much, ma'am. Let's quickly take a Q&A session, ma'am.
Hello. Yeah, good morning, ma'am.
Yeah, good morning.
Ma'am, this is with regard to diagnosis, ma'am. So recently, when we observed RT-PCR result of Omicron, so there have been, various gene detections, ma'am, like S gene and ORF reading frame detection and all. But how to identify whether it is Delta variant or, Omicron variant based on RT-PCR report?
Well, current commercial kit that we are using, it's literally not possible to identify exactly pinpointly whether it is Delta or any other variant. But yes, as I said, the S target is been missed. So in absence, it's indirect. So absence of S can give us an direction that probably this is due to Omicron, as because there is only, if I talk about my country, there is Delta and Omicron only. There is no Beta or Alpha right this moment. So S gene mutation is mainly present in Omicron. So that is how we can have a preliminary idea, but still to confirm that we need to go for sequencing.
Ma'am, you mean to say, like, if S gene is detected, it can be a Delta variant, and the absence of S is Omicron?
Exactly. Okay. Right this moment.
Yeah.
Thank you, ma'am. There was a question in the chat box. Is it necessary to go for vaccination for a 70-year-old person? Or is there any alternative to protect them from Omicron? That was the question, ma'am.
Well, for a 72-year-old, it is rather mandatory to go for the vaccination or boostering. And first thing you should do for that person to protect from all this is vaccination. As I said, the world epidemiology already has shown that the death because of Omicron is very less, but whoever have died, they have died. I mean, the history says they are not either vaccinated or they are overburdened with many other comorbidities, and they are quite aged. So please make sure that 72 years person get vaccinated.
Excuse me. Yeah, if you see COVID infection, right from the first variant to recent Omicron, why it has spared certain age groups. Like initially first stage, they said it's above 60 are more affected. Afterwards, more than 40 age are affected. Next, now they are saying Omicron. Before, recent one, they said kids are more prone for this infection. Is that because those groups were earlier, after infecting, they were vaccinated and they are spared, or is there any other reason?
Well, these are all hypotheses, but as per my understanding, it is like initially when it came, it was in one shape. So later after, more we are pressing them, more we are trying to fight with them, they are changing. So these viruses are continuously changing. So wherever they are finding obstacles, they are trying to change themselves to adapt with the situation. So initially, the variants, they could only infect the people with lower immunity, like high old ages, not the younger, as it was said that the receptors are less for the younger one. So with the time, what we saw that the next generation variants, they gained that virulences to overcome the immunity of younger generations. So they could fight back and they could effectively infect the, especially Delta. So 40, 50 years, everyone was infected. So now, and also because of vaccinations, the senior citizens or this, I mean, initially the vaccine was approved for 18 and above. So those group has got some kind of immunity inside. And mostly during the lockdown time, whoever had to go out, mostly this active group of people, kids, they were all back at home. So they remain mostly unexposed, and there is no immunity for them. Many of them does not have. So now what we can see that these groups are already got infected or vaccinated. So the group left are the children. So yeah, obvious. It is quite obvious that the affected group this time will be the kids, obviously. So like that, the virus evolution is like they will be changing again and again to adapt with the adverse situation.
Thank you.
Thank you, ma'am, for patiently taking up all our questions. To propose the vote of thanks, I would like to invite Dr. D. Madhusudan Reddy, sir, Director IQAC and Coordinator of the conference. Over to you, sir.
Yeah, thank you. Dr. Rukhsana Rahan, Associate Professor, Department of Microbiology, US Bangla Medical College, Bangladesh. Really, we are very happy to have you here for this session, ma'am. You have enlightened us on global statistics on COVID-19 and its structure very elaborately, and how mutations leads to various COVID variants, like right from alpha to Omicron. And you have thrown light on sites of mutations, especially in receptor binding protein region of spike proteins, which has led to many mutations and many variants till today, whatever in the environment. And vaccines for protecting these COVID-19 vaccine, COVID-19 infection. Detailed information of development of various vaccines during spike protein with the help of spike protein information, and effectiveness of these vaccines against variants, especially as per your presentation, we have come to know that Pfizer is most effective when compared to other variants, other vaccines when it comes to controlling these variants. And one more interesting thing is after 21 days of first dose, it was almost from 37.4% to 9.9% mortality rate has been decreased. And especially after second dose, after 21 days, that has come down to 0.8%. This is really, I mean, that shows vaccination, the importance of vaccination. And no vaccine can stop spread. Yes, that is true, because it is our preventive measures like wearing masks, maintaining physical distance and all will contain this virus spread. And really, we are very thankful to you, ma'am, for spending your valuable time. Thanks a lot.
Thank you, sir. Ma'am, please accept our certificate of appreciation. Thank you so much, ma'am.
Thank you all very much for hearing me with patience, and I'm really glad that I am here in this platform. If I could help anyway, then it's my pleasure. Thank you very much.
Thank you, ma'am.
Rukhsana ma'am, thank you very much for your valuable time for us. Thank you, ma'am.
Thank you. Thank you. I am honored.
Thank you, ma'am.
I thank all the participants for cooperating with us all through the technical sessions. Now we have a souvenir release by our chief guest and chief patron of the conference, Professor L.B. Lakshmikant Rathod sir, Vice Chancellor of Palamuri University. I request our registrar sir, HOD Department of Microbiology and the whole Department of Microbiology to join our chief guest for the souvenir release. Shall we all clap virtually for the souvenir release? Thank you, one and all. Next, we would like to take the feedback from the participants. Any participant
Thank
who are
you very
interested?
much. Thank you very much to everyone. We will share you the souvenir soft copy to each and every one participant. Thank you.
It's a feedback time from the participants. Any participant can raise your hand to share your experience. Any participant who are willing to share their feedback regarding the webinar can please raise your hand. So that we can unmute you.
Now participants can directly unmute and ask the questions. Feedback, if anybody want to give the feedback.
Yes, Chandana, please unmute.
Hello everyone, to all the respective madams present here and even sirs. I am Chandana from Telangana, Hyderabad. Actually, I am not at all a science background.
Chandana, you want to speak something?
Yes, sir.
Yes, she started speaking, sir. Carry on, Chandana.
Yeah, ma'am. I am not at all a science background student. I am a commerce. I have a commerce background. Hello, ma'am.
Carry on.
Yeah, ma'am. Someone was like they muted me, ma'am. So Yeah, ma'am. So, even out of curiosity and like I am very much interested in science field. So, due to some issue, I couldn't go into science field. But out of this enthusiasm, I always wanted to participate in all these science activities. So, I thank all the members of Parliament University of Microbiology Department for giving us this opportunity. All go through from Karnataka to Telangana. So when this PDF for the brochure reached the college, I was the first person who just enrolled into the
Michael
form.
Chandana, you want to speak?
Yeah, ma'am. Yes, sir.
She is speaking, sir.
You are unmuted for that only, or?
She's speaking, sir. We are able to hear her.
Okay.
Continue, Chandana.
Yeah, ma'am. Anybody can they see me? Can
Yeah.
you see me? Yeah.
Yeah.
So due to some issues, I cannot go into science field.
Your
but
audio is having problem.
Is that to everyone present over there?
No, we are able to hear you.
Oh, okay. I think sir has having any network issue from his end. Yeah, ma'am. So thank you. I thank each and every member present there. Actually, I was very much interested in biology, even though it is zoology or botany, anything, but I'm very much interested.
Unmute Chandana. Unmute yourself.
Thank you.
Chandana, please unmute and continue, ma.
She
But
finished it.
someone anybody ma'am, someone said me to mute myself.
You're done with your talk?
No, ma'am. Just I wanted to share my experience from this 2-day program.
Yeah.
Can I, ma'am?
Yeah.
Yeah. Thank you, ma'am. So, in this 2-day program, I thought that only there will be some discussion between the delegates and the members, not between the students. Or I didn't expect that there will be a question and answer among the delegates and the participants. So when a speaker was you gave us permission to ask the questions, and when I asked and my doubts regarding this COVID and etc., the Omicron, everything was being cleared, and I really enjoyed it. I fetched lot of knowledge from this, and even I understood how depth the science field is all about. I thought only it's relating to till what I studied. Okay, it's that I thought it will be in depth, but not to this extent about the microbiology and etc. Yesterday, when I saw many doctorates were talking regarding so many matters, when the speakers are many questions were raised, and they were even giving the main Dr. Anjali ma'am, I think, and many more respected delegates were giving so much of information how to present a PPT, and they even suggested so many ladies who are pursuing a PhD, and even who are trying to go for PhD or doing master degree. So I really appreciate and I am honored, and I'm very happy that I'm a student who from commerce field and participated in the science field, and I loved to just join here. And among the other participants and delegates, I hope I am the only student present over here. I'm yeah, ma'am. When I saw the participants list here, everyone was when yesterday everyone were presenting, they were all doctorates and MSc, etc. I was literally afraid that I'm only a student over here. I'm very happy that I'm here, ma'am. I'm totally overwhelmed. Thank you so much for giving this opportunity. Yeah, ma'am. I now I'm simply regretting I didn't go for science field when I just enjoyed this 2-day program. Yeah, thank you so much, ma'am. Thank you, ma'am.
Thank you, Chandana.
Chandana, this is also for the platform for students also. Mostly for students. We don't feel like doctorate and all.
Yeah, ma'am. That's why when yesterday,
We appreciate you.
when I saw, like, I was thought like I got so much of information, and even like how to present a PPT, how to talk, how to answer when a person is asking something to you, etc. Ma'am, like I really enjoyed this today. Thank you for actually thank you for coming up with this great international e-conference idea, ma'am. I thank all of them.
Thank you, Chandana. So good to hear from
Yeah,
you.
and I'm a native of Karnataka only, ma'am. My mother tongue is Kannada only. And additional information. Yeah, thank you, ma'am.
Okay. Next, we enter into the valedictory session. I invite Dr. Madhusudan Reddy, sir, to welcome the guests. Over to you, sir. Dr. Madhusudan Reddy, sir.
Hello. Hello.
Yes, sir, you're audible.
Yeah. I'm sorry. Yeah. So it's my pleasure to start this valedictory function. So with most privilege, I welcome Professor Arvind Deshmukh, sir, President MSI, and Dr. K. Ratna, Hyderabad chapter, of Indian Women Scientists Association, IWSA. And I think we have got another guest, Professor Arvind, Deshmukh sir. He will take care of, I think, MSI awards. Hello, I'm able to. Hello, can you hear me, ma?
Yes, sir.
Yes, sir, you're audible, sir. Carry on.
Yeah. So really, so yesterday it started, and we had 2 sessions yesterday and today morning session. So everything went on well. So acknowledging the participants, and most respects to all the dignitaries who have spent their valuable time with us. With more privilege, I welcome all the chief guests and guests for this valedictory session. Thank you, ma'am. Hello.
Thank you, sir, for welcoming the guest. I request to Vasavi madam, please give introduction about the Deshmukh sir.
Yeah. Thank you, Dr. Shanti Priya. I am privileged to introduce Professor Deshmukh Arvind Madhavrao, President, Microbial Society, India. Professor Deshmukh is an ex-professor and head Department of Microbiology at Dr. Babasaheb Ambedkar Marathwada University, Osmanabad. His research areas are nanotechnology, biotechnological solution to e-waste, biodiversity of actinomycetes, biogas, and dye degradation. He has a teaching experience of 37 years and published 82 research papers, of which 32 are in international journals. 17 students were awarded PhD under his able guidance, and 1 is working. Professor Deshmukh has 1 major and 2 minor research projects. He has written 4 books and edited 7, organized 46 state, international, national level conferences, seminars, workshops in India and abroad. He is the chief editor of the Journal of Microbial World. Professor Deshmukh worked as a chairman, Board of Studies in Microbiology, member of Board of Studies Research and Recognition Committee, Board of University Teaching and Research Committee, Environmental Committee of many universities and the government. He was a resource person in 154 conferences and 285 colleges or universities, and gave 14 talks on creating awareness on biofertilizers, biopesticides, pure water, vermicompost, and environmental pollution. He has written 20 scientific articles on biotechnology in Marathi newspaper. Sir has visited various countries like Japan, Malaysia, Nepal, Bangladesh, Thailand, Indonesia, Dubai, Singapore, and Bangkok for paper presentations. He was an invited speaker in conferences held at Mauritius, Bhutan, Sri Lanka, Bangladesh, Nepal. Professor Deshmukh visited universities or institutes at USA, China, Germany, France, and United Kingdom out of his curiosity in science. He has organized international conferences at Karad, Aurangabad, Sri Lanka, Bhutan, Mauritius, Dubai, USA, and Andaman. Sir has been conducting virtual classes in microbiology from 5 years, where lectures of eminent scientists from India and abroad are arranged. Thousands of students are taking advantage of Sir's lectures. You can visit him at the websites allmicrobes.com and microbiosociety.com. Thank you for giving me this opportunity.
Thank you, sir, for joining us for the valedictory.
Thank you. Thank you,
We
madam.
also invite our next guest of honor, Dr. C.B. Lakshmi, who is a scientist at IICT. Dr. C.B. Lakshmi, she completed her graduation and post-graduation in chemical engineering from the College of Technology, Osmania University, Hyderabad. She started her career in teaching at Loyola Academy, Secunderabad, but later moved to Regional Research Center, which is currently called as IICT, Indian Institute of Chemical Technology, Hyderabad, as Scientist B in Chemical Engineering Division. In June 2002, she became the head of the Research Management Division. As part of the RMA activity, she handled the project management, HR management, international S&T affairs, including foreign deputations, IPR management, institutional events management, conference funding, institutional awards, financial management, and institutional repository. Thank you, ma'am, for joining us for the valedictory. I feel privileged to introduce ma'am. Thank you.
Please welcome, ma'am. I request Deshmukh sir, please give your message for this session.
Okay. Thank you, ma'am.
Sir, please, sir, continue.
Thank you. Really, I'm very happy today. The reason is that from today morning, I'm watching your event. And excellent speakers in the field. And one more thing, the topic of this conference is a need of today's time. Because everyone is worried about the pathogenesis, pathology, and particularly Omicron virus. And everyone wants to know if it is a virulent, whether all answers have been given by the different speakers. I really appreciate the efforts taken by Shanti Priya ma'am. She was regularly in touch with me. And with a continuous efforts, she made the event successful event. Many presentations were there in the conference. And it is really a feast to the academic area. The students who are interested in academics, they have enjoyed. Just one student from commerce also has expressed his feelings regarding the conference. As a president of Microbiology Society, I assure you, and it is my duty to promote, to support such activities, not only now, but even in the future. Whenever you are doing the best in microbiology or biotechnology, It is my duty to support you by all the ways and all the means. And honestly, I'm doing it. So, as a president of Microbiology Society, I want to congratulate the team because only Shanti Priya ma'am cannot do it alone. She needs a team support, and your institute, your honorable principal, they are successful in creating academic environment in your college. As a result of it, you are organizing such beautiful events. So again, congratulations to you, ma'am, and all your team for successfully organizing such conference. And thank you very much for calling me, for giving me an opportunity in the valedictory session of your 2 days continuous efforts. I'm very happy, and I wish all the best to all participants also. They have attended sincerely. They gain much knowledge during these 2 days. Now my request to all participants is that to percolate this knowledge, to transfer this to your college students, to your friends, to other teachers. Thank you, ma'am, again. Thank you very much.
Thank you. Thank you so much, sir, for your support and collaboration with us for this e-conference. Thank you so much, sir, for your valuable words regarding us.
Thank you, sir. A small announcement to all the participants. All the registered participants will receive their participation certificates. A hard copy will be sent to your address. Thank you. Hard copy of the certificate will be sent to you, to all the registered participants. Thank you. So as we all know, yesterday we had paper presentations and poster presentations.
Mercy, 1 minute.
Yes.
I request to CB Lakshmi ma'am, please. Professor Lakshmi ma'am, please share your words with us.
Yeah, sure, Shanti Priya.
Ma'am, please welcome, ma'am.
Yeah, thank you so much. I'm in fact very elated and honored to be part of this. I hope you're able to hear me.
Yes, ma'am. Yes, ma'am. Please.
Yeah, I'm very honored and elated to be part of this professional gathering. Let me on the onset congratulate and appreciate all the organizers, a teamwork which has excellently been performed these 2 days. And it is a great initiative by all of you to think of a topic which is a very hot topic, in and out every day we are discussing, whether it is educated or not or non-educated. Only topic we talk about is in a day is COVID, vaccines, the repercussions, the symptoms, the sufferings, all this. Anyway, I'm very happy to say that you organizers have very well planned all the sessions, and you have selected very good speakers who have covered almost all the aspects of COVID. A lay person also is able to understand the various scientific sessions, which have been scientific at the same time. They are on the popular lecture mode. I congratulate all the speakers of both the days, and I'm very happy that I am given an opportunity to say a few words on this occasion. Let me thank Palomar University and also Microbiological Society of India that you have thought of collaborating with Indian Women Scientists Association Hyderabad branch to be part of this very important scientific session. Thank you so much. On behalf of IPSA, I once again thank all of you, the organizers, for involving us. And now, without going into the details of the lectures or about the speakers, since we have heard a lot about the speakers and also the scientific content they presented in the various sessions, I'm not competent enough to technically or scientifically comment about their work, but I really enjoyed all the sessions. And being in applied sciences field, my focus generally would be on applied sciences and technology field. My area of interest have always been technology forecasting, technology demonstration, applied sciences, product development or process development, and taking the technology or science into the field. As part of IPSA, we are trying to create interest among the youngsters to take to sciences and take science to the masses in general. See, science is everywhere, all around us, within us. And let us all realize that science is very, very vital and important in our day-to-day life. So keeping this in mind, I have a message to all the youngsters who have presented papers, who have actively participated in this conference, mainly to request the Palmer University management to focus on applied sciences, especially about the innovation, entrepreneurship, and science demonstrations. Please give them a platform and please make your students aware of the various facilities, careers, and the opportunities that are available to them in the present situation, where even the government and private partners are playing a major role in coming up with startups and incubators. Especially in your field, this life sciences incubator is there in Hyderabad, that is the IKP Knowledge Park, which is totally a life sciences incubator. Please make your students aware of this and make them, let them go on a virtual tour of what actually an incubator is and in what way their scientific outputs can be utilized and be taken to the field and to the society. And unfortunately, we are very proud being in India that we have a woman biotech incubator park in Chennai that is specifically only to the women. And please make all these aware to your students so that their interest will be created. It is not just after their PG or after their PhD. It is not just taking a 9-to-5 job. There are so many other opportunities available to them. And I think you people should make a platform for them to make aware of this. And we as IPSA, we can definitely help you out in case if you are interested. We have been in contact with all these S&T parks, technology parks. In fact, IICT itself was sometime in the late 90s, we have set up a biotech incubator in Shapurji Pallanji Park in Hyderabad. And that has been initiated in a very primitive stage initially, and now it has been working very well. There are very number of people who have set up their incubators and they are working, and they have set up their commercial unit subsequently after incubating their scientific ideas. And this is a very important aspect which you people should, though I don't know whether it is included in your curriculum or not, but make it a point to have it as a optional or a specialized subject. You can make them aware by means of lectures, demonstrations, workshops, visits, virtual tours, and we can with Palamuri University being a budding university, we can handhold with you and we can help you in promoting this in your campus. And another aspect which I'm very much interested to promote is the product development. That is very, very important. And India is not very good at product development. And please ensure that your students are the orientation program for such societal important projects and programs. And also a message to the faculty is that I would request all of you to understand the opportunities that are available in taking up projects in the campus. Especially CSR helps all the youngsters, young scientists and professors and teachers, science teachers with a project mode up to around 5 to 10 lakhs. They give seed funding to work on projects, and that gives you the confidence in developing a project or process or in a project mode. Please take advantage of these opportunities and avenues available. DST, Department of Science and Technology, Department of Biotechnology, all these departments are working towards the interaction between the academia and also research institutes and industry. This collaboration should be developed and established so that your students will come out with flying colors and they will be known globally also with their scientific innovations and ideas. And I would like to add one more thing, that we can have another program, especially for the students, as a workshop or a lecture mode, a program on scientific innovation and entrepreneurship and about the startups. We can have visits to various industries and startups also during this program. I volunteered this to the Palamur University and also all those institutes representatives who are present on this platform. With these few words, I just congratulate all of you once again and congratulate all those winners. Everyone is a winner who has made a presentation, who has put up a poster, an initiative that has been taken by you, and a platform which has been given to you, which you have taken advantage of. Thank you so much. Thank you, Shantipriya and Sridevi, in particular, for roping in to this Palamur University platform. Thank you one and all.
Thank you so much, ma'am, for your valuable words and suggestions. We will definitely follow that. Thank you, ma'am, for your valuable words.
Thank you.
Thank you so much.
Yeah, my pleasure.
Over to Mercy.
Thank you, ma'am. So to announce the best paper presentation, I would like to invite Professor Arvind Deshmukh, President MSI, and Dr. N. Kishore, HOD, Department of Microbiology, Palamuru University. Over to you, sir.
Thank you. Thank you, madam. I'm very happy because many oral presentations were there. Many poster presentations were there. So first of all, I'm happy to declare oral prizes. Prizes for oral presentation. So first prize goes to Usha Rani. Oral number 09, Usha Rani. Second oral prize goes to Raghavendra 08. And a third oral presentation prize goes to Pavan Kumar 07. I congratulate these three candidates. Along with them, consolation prize is there for oral presentation. Consolation prize goes to Revati and Pratibha and Vrashali. So congratulate all of them for excellent oral presentation in the present conference. Lastly, poster presentation. Poster presentation prize goes to Mercy Anitha Rani and M. Mahesh. Poster number 07.
Okay.
So, on behalf of President, as a President of Microbiologist Society India, I appreciate all these winners. Also appreciate other participants, although they could not get the prize, they have participated. Congratulations to the winners and appreciate the efforts of non-winners. Thank you very much.
Sir, thank you so much, sir. Kishore sir, can you repeat that results again once more? Audience are asking participants.
They are with you, madam.
Sir, could you please repeat the Deshmukh sir, please.
I think we can share in the WhatsApp group later.
Yes, we will share. Thank you so much, sir, Arvind Deshmukh sir, for announcing the results and putting inputs for us. Thank you so much, sir. Now the
Thank you, sir. I would like to invite Dr. Shantipriya Ajmera ma'am to propose the vote of thanks. Over to you, ma'am.
Good afternoon, everyone. Honorable Chief Guest, Vice Chancellor Sir Professor B. Lakshmikant Rathod Garu, respected Registrar Pavan Kumar Sir, Principal Campus PG College Dr. Noorjahan Ma'am, Dr. Madhusudan Reddy Sir, OST and Director of IQAC and Coordinator of this program, Dr. N. Kishore, HOD of the Department of Microbiology and co-convener of this event. I am very much thankful to you for your able guidance and support to conduct this event successfully. I am sincerely thankful to respected Professor Arvind Deshmukh sir, National President of MSI, and I am very much thankful to beloved convener Dr. K. Ratna ma'am and co-convener of the Indian Women Scientist Association Hyderabad chapter, C.B. Lakshmi ma'am. I am very blessed with your collaboration for this event, both MSI and Indian Women Scientist Association. I am very thankful to you for your support.
అక్కడ మాట్లాడేది ఇక్కడ వస్తుంది ఇందులో వస్తుంది అలా వీసి ఛాంబర్లో
Convener, I am very happy as a convener and organizing secretary of this two-day international e-conference on microbial pathogenesis and recent advances in diagnosis of Omicron, with association with Indian Women Scientist Association and MSI and IQAC, Palamur University. I would express my gratitude to all for their support towards the successfully organizing this conference. I especially thankful to our speakers. First day, Professor Anjali Devi ma'am as a keynote addressee. She shared about the good and bad microorganisms as well as nutrition and about the innate immunity. I am very thankful to ma'am. And the second speaker, technical session, first speaker Professor Sharmista Banerjee ma'am from HCU. She shared and a detailed talk about the tuberculosis and the metabolic adaptations of Mycobacterium tuberculosis, and the drug resistance and adaptive responses, and about the Shikimate pathway. I am very glad about ma'am talk. Thank you very much, ma'am. Dr. Gokul from Malaysia. He given his lecture on mycotic infections in detail. Thank you so much, sir. Second day, as well as second day, the speakers, Professor DVR Sai Gopal sir, Vice Chancellor of Cluster University, Kurnool. Sir was shared about the variants of coronaviruses and mutations of that variants and advanced viral diagnosis and various immunoassays. I am very thankful to you, sir, and honored with your presentations. Thank you very much. And another fourth speaker, Dr. Rukhsana Rahan, Associate Professor in the Microbiology Department of US Bangla Medical College from Bangladesh. Ma'am was shared about the SARS-CoV-2 variants and various vaccines in detail and explained the reason why Omicron do not infect the lungs. I am thankful to you, ma'am. So coming to the jury members. The panel members of the paper presentations, Professor Archana Giri ma'am from JNTU Biotechnology Department, Dr. Sandipta ma'am, Usmania University from Microbiology Department, and Dr. Ramchandra Merbu, Assistant Professor from Mahatma Gandhi University, Dr. Sai Padma ma'am from Bhavan's College, Autonomous College. I am very thankful for your valuable time and justification of the presentees. I would also like to express my gratitude to all the delegates and my dear participants for your presence and contribution to make this e-conference a successful. I hope all of us have gained good knowledge from this program. Last but not least, I would like to thank organizing team and technical team and electrical team, especially Dr. Arjun and Dr. Anjaneyulu and Gautam for their technical support. Really, with your support only, with your help only, the program ran very smoothly. I am very thankful to you both. And I'm looking forward to conduct many such events and to be helpful for the microbiology fraternity and university development. Thank you one and all who are participated in this event. Thank you so much. And I request you all to give a feedback. And last but not least, I would like to thank the press and media with us for their cooperation. Thank you so much. Over to Miss.
We thank all the dignitaries, all the jury members, and all the participants for actively participating and cooperating with us all through the webinar these 2 days. Thank you very much. Expecting the same cooperation further. Thank you so much.
We shared the feedback link in the chat box. Please, everyone, please, you share your feedback there. Fill that feedback form and send. Then only you can receive that hard copy of certificate.
Note to all the participants, please fill the feedback form which is provided in the chat box.
So I am also thankful to Dr. Vasavi madam, Dr. Sridevi ma'am for your support. Anyone I missed, please don't take the mind, please. Thank you so much for all. Merci. Thank you so much for your cooperation and supporting me. Mrudula
Thank you,
madam
ma'am.
from
Thank
the
you so much.
department. Merci. She is my research scholar, my student, as well as assistant professor in the degree college. Thank you very much, Merci.
Thank you, ma'am. It's my pleasure. Thank you, ma'am. Thank you, one and all. If you have filled the form, you can please exit the webinar. Thank you all once again for joining. Have a good day.